Bem, boa tarde a todos. Eu saúdo a todos e convido, para assistirem o segundo webinário do projeto CAPES Print da UFC, que está ocorrendo durante os encontros universitários de 2021 da nossa universidade. Esse é um evento que é uma atividade promovida pela Pró-Reitoria de Pesquisa e Pós-Graduação, pela ProInter e pelo projeto intitulado Engenharia de Tecidos e Microfluídica no Controle de Doenças Orais Biofilm Dependentes e suas implicações orais e sistêmicas, que é a parte integrante do projeto CAPES Print, que é o nosso projeto de institucionalização, da UFC. Esse projeto é um dos 17 projetos que compõem o projeto institucional da UFC, e tem como parte integrante professores tanto do programa de pós-graduação em odontologia, principalmente, e microbiologia médica. Farão parte desse evento os nossos parceiros no exterior, a professora Simone Duarte, da Indiana School of Dentistry, o professor Luiz Bertassoni, da Oregon Health and Science University School of Dentistry, e o professor Alex Mira, da Foundation for the Promotion of Health and Biomedical Research of Valencia Region. Esse webinário terá uma apresentação de 40 minutos de cada um dos participantes em inglês, começando pela professora Simone Duarte, Ladies First, depois o professor Luiz Bertassoni, e depois será finalizado pelo professor Alex Mira. E após essas 3 apresentações, a gente vai fazer as perguntas que podem ser feitas pelo chat no YouTube, está disponível para vocês. As perguntas podem ser feitas em inglês ou em português, e elas são selecionadas, caso sejam necessárias, e feitas através da coordenadora, do programa de pós-graduação do odontologia, a professora Lidiany, que está aqui presente. E agora eu também chamo o professor Rodrigo Rego, que faz parte do nosso programa em odontologia e é coordenador da ProInter, para fazer a apresentação dos nossos palestrantes. Antes disso, eu gostaria de apenas um agradecimento especial ao professor Alex Mira, que ele ainda não faz parte do nosso projeto Print, mas é um parceiro, um colaborador em pesquisa bastante importante para nós aqui. Muito obrigada, professor Alex, e obrigada a todos. Aproveitem.
So let's change to English. Good afternoon. So I'd like to thank you, Professor Lidiany, and also Professor Cristiane Fonteles, who is Professor Lidiany is the Dean of the Faculty of Pharmacy, Dentistry and Nursing from the Federal University of Ceará, and also Dr. Cristiane Fonteles, who is the coordinator of the graduate program in dentistry. But today we have special guests, so I'd like to thank Professor Simone Duarte, Professor Luiz Bertassoni, and Professor Alex Mira. So, as Professor Lidiany said, this is a very important project for us at UFC, actually in Brazil. It's the CAPES Foundation is sponsoring a big project. So, UFC is among public universities engaged in this project. So, today we are going to have the presentation. As Professor Lidiany said, we are going to have the Professor Simone Duarte, who is from the Indiana University School of Dentistry from the United States. Professor Luiz Bertassoni from Oregon Health and Science University School of Dentistry, also from the United States. And we have another guest, is Professor Alex Mira from the Center for Advanced Research in Public Health, and from the Fundación para el Fomento de la Investigación Sanitaria y Biomédica de la Comunitat Valenciana from Valencia, Espanha. We have, I know that we have in different times here. We have Alex, thanks a lot. I know it's night for you, and thanks Simone and Bertassoni, you are a little bit behind us, so we found a time between everybody so I hope we can manage it well. So please, the first guest to talk is Professor Simone Duarte. So it's nice to see you again. It's been a while. The last time I saw you, we were together in New York City. Now you are in Indiana. Professor Simone has been collaborating with our graduate program for a long time. I'm sorry, I know we have a strong collaboration with us, so I'd like to thank you again for this. And please, you can present. And after that, like Professor Lidiany said, we're going to have the 3 presentations, and at the end, we're going to have the discussions about the presentation. So thanks again for you all, and thanks everyone who is watching us on YouTube. So please, Professor Simone Duarte.
Thank you. Thank you, Rodrigo, for the kind presentation. It's nice to see you again as well. So let me share my screen. Okay, I hope you guys are able to just give me a minute. We practice, right? But it's not working. Okay. Are you guys able to see it?
Not yet.
Not yet. Sorry. How about now?
We can see, but it seems a little. Okay, it's okay now.
Is it okay? Okay, I just want to remove this. Okay. Okay, are we good? So thank you again for the kind introduction. I want to thank the organizers for this beautiful event. I'm very honored to be able to share a little bit of my research, my understanding on oral biofilm research, and again, very pleased and very happy. And sharing the stage with this brilliant scientist, it's really an honor. So I'm Simone Duarte. I am from Brazil. I graduated, I did my DDS, Master and PhD at FOP Unicamp in Piracicaba, where I am actually from. I was born there. And as part of my PhD, I did a sandwich program from CAPES at University of Rochester, New York, in the United States. I came back to Rochester to do a postdoc, and then I went to NYU, where I worked for 10 years as a professor. That's where I met Rodrigo. And since 2017, I joined IUSD, Indiana University School of Dentistry. So I've been here for about 4 years now. I'm going to talk a little bit about biofilm and oral biofilm research. Before that, I want to introduce a little bit of my understanding on biofilm to you guys. So when we talk about microorganisms, it's important to remember that they are part of us almost. So we have a huge number of microorganisms in our body, what we call human microbiota. They are there and we want them there. We actually need them there because they're part of our system. They are very linked to our health. Probably the biggest example is the gastrointestinal tract microbiota, the GIT, or intestinal flora, intestinal microbiota. That is, it helps us in so many ways. It helps us with nutrients from digestion. It helps us with metabolism of drugs. It helps us with vitamin, like vitamin K is completely dependent on the GIT microbiota, and mouth is part of the GIT. So that is important for us to keep in mind. So every time we are going to mass up with this biofilms that we have in our body, we have to be extremely careful because they are actually part of us. We actually have 10 times more microbial cells in our body than mammalian cells. I joke that we are just a big scaffold for all this microorganisms to live in. So biofilms are usually the way that these microorganisms are organized, are communities actually that are extremely successful. They can live and survive and thrive in very extreme environments and conditions. They are very resistant to treatment, to environmental challenges. So they are really capable of organize themselves in a way that they can survive. And that is something that we have to really understand to be able to do research in biofilms. So why we should bother with biofilms? In general, I just gave you an example of our own body and a biofilm that lives in it and that is important to us. So in general, they're good actually. We also use biofilms in several different situations like bioreactors. We use biofilms to treat waste in water, environmental cleanup. So there are several ways that we can actually use biofilms as part of our ecosystem, just like any other living being in the ecosystem. But of course, there are the bad biofilms where usually the focus go to, right? Bad biofilm, it can cause corrosion, fouling. We have industry that closed down because of contamination in equipment. We have hospitals that closed down because of contamination in water system. So that is a big issue. And of course, the one we talk about all the time, that is the biofilm that causes disease. Some causing infection, some causes dysbiosis, different types of diseases. And the issue then is that this bad biofilm, including all this equipment, industry, and everything, it has a huge economical value. So over billions of dollars are spent every year due to contamination with biofilm, either in our body or industries, and even like spacecraft had to come back to the planet because of contamination in the water. So just to give you an idea, usually biofilms, they follow the same, a similar pathway of formation, growth, and development, maturation. In general, we have single cells that will bind to a surface. Once they bind to the surface, the single cells, we call them planktonic cells, free-floating cells. So they bind to a surface, and because they have affinity for it, either cell membrane with a protein receptor in the membrane of the or in the surface, they bind to it, and then they start to produce this extracellular polysaccharide, which is almost like a glue. So they start to glue themselves in this surface, and then as they produce more matrix, other cells come and they become more mature biofilm until they reach a stage where we call it actually mature biofilm. In this diagram here would be stage 4 or 5, where there are even changes in the bacterial phenotype. Probably because they don't have to spend a lot of energy anymore surviving, because they have this barrier that is this extracellular polysaccharide matrix that will kind of help them to survive in that environment. So their metabolism will become very low. The cell division is also very low because they don't really need to. They don't want to grow too much actually anymore, because that can that will mean competition, right, for nutrients, etc. So when it reaches this stage, this changes in phenotype, they are important mostly when we look at ways to kill the cells. A lot of the antibiotics we have, they actually work during cell division, or they work in specific enzymes as part of the metabolism. So in these mature biofilms, they are just not going to work. Another characteristic of biofilm that we really have to pay attention is that as I mentioned, it's a community. So as part of a community, we have the microorganisms that live there, but we also have this whole structure that is around it that has several properties that we are learning more and more. That's what we call the extracellular matrix. So we know a few of them. For a long time, we just focused on the bacterial cells or microorganisms that were there, but now we are learning more and more about this matrix and what it does. Take it like, almost like, if we think about our community, right? We have the houses, we have the streets, we have internet, that we can communicate. It's almost like that, but in a micro situation. So in the matrix, the quorum sensing, I don't know if you're familiar with this term, but quorum sensing is communication. It's almost like the internet in this case. So bacteria that are in one side of the biofilm, they notice a change in the environment, for example, a decrease in the pH, and then they release signals, chemical signals, that will travel through this matrix to other parts of the biofilm where the pH is not necessarily low yet. But these cells will notice that the pH might drop, and they already start to express genes that will help them to protect themselves in the low pH. So it's very complex the way they can communicate. They also release enzymes in this environment that can help them. For example, the penicillin group, it's an antibiotic that is called beta-lactamic, right? It's the structure of the antibiotic. And there are some bacterial cells that are able to produce an enzyme that's called beta-lactamase that breaks down penicillin group antibiotics. Once we take penicillin, this bacteria will release this enzyme to the environment. So if we have a matrix there, the enzyme will almost form like a dome. And when penicillin penetrates, it will be broken by this enzyme. So in this dome, whatever is in there is protected, even sensitive cells. So they kind of use that structure and that organization to really survive. There is a lot of competition there as well. But again, if they have to protect themselves from external challenges, they'll do it in a very good way. Dental biofilm, as any other biofilm, has a very similar structure. Again, it's very complex, either structurally and dynamically. And if this polysaccharide matrix, just as I mentioned in the previous slide, it also has these properties that are there to make this biofilm survive to extreme environmental challenges. So if we were able to disrupt this polysaccharide matrix, probably we would be able to reduce this tooth decay. And I will discuss that a little bit more, okay? So we don't know much, though, about the physical properties and how this matrix works. We are learning more and more and trying to understand, but we do know that the cells in biofilm, just like any other cells, when they become mature, which means usually bulky, we have low metabolism, we have low cell division, and we have a lot of quorum sensing communication. So those are some of the virulence factors that we call when a dental biofilm becomes a mature biofilm, and in general, carcinogenic biofilm. There are over 700 species of different species of microorganisms in the biofilm. I'm not going to go into details here. We have an expert that will talk right after me. Dr. Mira, he will probably be talking in a lot more detail and with a lot more knowledge than I have in the species that inhabited this biofilm. But there is one where in which I am 100% sure everyone that heard about dental biofilm and caries has heard of, which is the Streptococcus mutans. For a long time, it was considered the cause of caries. People were looking at number of Streptococcus mutans to see if that patient was in high risk of caries or not. We know that nowadays this is not really important. This is not really what causes caries. But there are some things that we learned about this microorganism because it is the most studied that we have, probably within all the microorganisms, even different in different fields. We know that this microorganism it produces these enzymes that are called glucosyltransferases. It's not the only one. It's not unique to as mutants, but they are able to produce these enzymes that they produce and they release. So the enzyme works extracellularly. And these enzymes, they break down sucrose. And the product is glucan, which is actually the main component of this extracellular polysaccharide matrix. So if there is sucrose and if there is GTF will break down the sucrose and we will have glucan, insoluble and soluble. Another thing that this microorganism has, again, not unique, and in this case, plenty of other microorganisms do the same, it is acidogenic, which means that it produces acid as part of the metabolism of sugars. Now, not sucrose, just sucrose anymore. Every sugar, basically. And then they produce acid and they're able to survive in this acidic environment. So what happens is that if we have a lot of sucrose, GTF that was released and works outside the cell, so it binds to water bacteria, it binds to salivary pellicle, even in solution in the saliva, we have GTF. Once we give this GTF sucrose, this enzyme will break down the sucrose from glucans, and that will help to make this biofilm bulkier. Once it's like biofilm is bulkier, saliva will have a hard time penetrating it. So that can be a huge problem because all the protection that saliva gives us will be actually not able to reach in the deep parts of the biofilm. I'll talk a little bit more about that now. So as I just mentioned, if we have sucrose, right? So sucrose is actually the determinant factor for caries for this specific disease, because sucrose is responsible for this bulky biofilm formation. Once I have the bulky biofilm formation here, let's say if we get this site here, so we have a bulky biofilm, and then we have the tooth surface here, and we have saliva that comes from the outside. A lot of sucrose, so the biofilm is bulky, and then saliva is not really able to penetrate. Bacteria that are here are producing acid from water-soluble glucans that they're breaking down, or from other sugars like even fructose or glucose, and they are producing acid. By producing acid, they are living this environment here in a low pH. Acidic environment, low pH, what happens is demineralization. So demineralization will, if not controlled, if not stopped, become caries. So that's basically how it works. Because there is this insoluble glucan here protecting this biofilm, saliva, which is a buffer that would bring the pH up, is not able to reach those deep parts of the biofilm. So you leave the acidic pH here for a much longer time, which means that the bacteria that are not able to survive in this acidic environment will die. So we have this shift in the microbiome, and that is a cycle that almost has no end unless we remove this biofilm from here. So acid, that comes from as a product of sugars, right? That are metabolized by bacteria are responsible for the demineralization. Common surfaces for caries are surfaces where we actually have this bulky biofilm. So that I'm talking about enamel caries here. So basically, excess of sugar is the determinant factor that will cause a stress, acid production, neutral pH, goes to low pH, mostly in that interface between the tooth and the biofilm. And then we have a microbial shift, which will keep producing acid and surviving in the acidic environment. And then we have an imbalance of remineralization, demineralization. Saliva, in addition to bring the pH up, is also responsible for the remineralization process. So again, just a reminder, dental caries, the determinant factor is sugar. This picture here I took at my pediatric dentist, my daughter's dentist here in Indiana. I went to her office. The first thing you see as soon as you come in is this picture of amount of sugar in your drink. I thought it was brilliant. I think every office should have that. Talking about oral biofilms, I talked a little bit about biofilms that are related to caries, but we also have biofilms that are related to other diseases, right, in our mouth. Periodontal disease is one of them. They're all connected and related in some way, but because the environment changes, the types of microorganisms that will be there and the resources they have will change as well. That doesn't mean they're causing a disease, but that means that the environment will collaborate and will have them surviving there for a long time, and that can result in a disease, not necessarily causing it. So when we talk about periodontal biofilm, again, I'm not going to go in details, but what we see, and this is the hypothesis that we mostly accept now, is that we have a plaque accumulation. There are specific bacteria that are present, so we see them when we have disease, again, not necessarily causing it, but usually when you have plaque accumulation, you have more inflammation because the body is just trying to protect that plaque, that biofilm, to go inside, right? To go into your bloodstream, which would cause a systemic infection. So by trying to prevent that biofilm from coming in, the body sends inflammatory response. When we have matrix, our immune cells are not able to phagocyte the cells that are inside this mature biofilm. They don't recognize it. Another issue. So they're able to phagocyte single cells, but not a biofilm because of matrix, which they don't recognize. So it keeps sending inflammatory cells to the point that it starts to absorb bone. So the body starts to reabsorb bone, trying to avoid that biofilm. And that's when we usually see microbial shift again and disease. Other microorganisms that we have in the mouth, so I'm just introducing them to you because I will talk about them a little later, are Candida. Candida, they're yeast, right? There are different types of cells. It's a eukaryotic cell. It's more complex. It has a nucleus. A lot more similar, even though it's not as, it doesn't produce as many ways for it to survive. It is a lot more similar to mammalian cells. Candida, it is what we call, it's a microorganism that we have there. And then if something happens and it can grow, it will cause a disease. So we have different types of Candida, and usually we don't see any problem people have in their mouth. However, when there is a surface where they can grow better, like prosthetics, for example, or if there is a decrease in the immune system, then this yeast will actually, they will be able to grow, and then that's when we see disease. So we do, we've been focusing on microbial cells, and most of the understanding that we have on metabolism and how the cells, they react to treatment and antibiotics, those are mostly known from culturing them in the lab in a way that they are planktonic or free-floating cells. But this is, again, not what happens. Even though we've been studying biofilm for a long, long time, there is still a lack of translation from biofilm research in the lab to biofilm research in the clinic. So that is something that we have really to enforce. So if you look at FDA, the way they look at antibiotics, the task they ask for is usually planktonic. So why? What can we do? So that's like when we have to really see and try to translate the research we do in the lab. So if we are doing biofilm research in the lab, it's very important that we understand and do a comprehensive evaluation. To do that, we have to ask the correct questions to find the correct answers. We just look for answers for our own questions, right? So it's very important to have a very well designed question to be able to understand. From that, we generate a hypothesis. We know how complex the biofilm is, so we can try to mimic whatever will help us answer that question in the lab. And of course, we will have several challenges, and it's important to try to recognize them before to be able to deal with them when they come. And then that's when we choose the methods. So I'm going to show a little bit now of the research I have been doing in my lab. And I hope you guys enjoy, and we can discuss that later. I'm just going to introduce them to you, and we can maybe discuss them later. There are different ways to test biofilms. A simple way is to look at the microorganisms that are there. So they will be different, right, in phenotype, as I mentioned. If you get a microorganism that is planktonic or in the biofilm, the same species, same strain, they will have different metabolism, different phenotype. So we can do that by culturing them, just seeing like, is it able to kill the cells, or let me recover the live cells from a biofilm when I treat with whatever treatment I'm testing. We can also do molecular biology. There is plenty of information on genome and microbiome of microorganisms. So that is, we have the techniques there, and we can actually look at genes they express. This picture here is the genome for S mutant. So we know every single gene that it has. But we can also look at the matrix, right? We can look at the types of polysaccharides that are there, the structure, the linkage, morphology, how porous it is, how rough it is. We can look at physical property, elasticity. We can look at the metabolites. Simple example is pH. Look at the acidic production by this biofilm, and so on. I test therapies in the lab. That's mostly what I do. However, as I mentioned, you should make the question first and then look for answers. So I usually try to understand what this therapy could be used for, and then I will choose a model and an assay that I can actually test this question. So here is one that I've been studying in different aspects. That is low temperature plasma. Plasma is the fourth state of matter. It is a ionized gas. So we have gas that go through a magnetic field, and then we have plasma as a result. It looks like a flame, but it's cold. And as a flame, it has some products that will go around it. And depending on the distance from the flame, these products will be different. Plasma is not new. There is use in industry for a long time. Those neon lights, they're made out of plasma. We have plasma in nature. The thunder is actually plasma. The interstellar environment is plasma. So we have like plasma, it's not a new thing. But plasma dentistry, though, is quite new. In Europe, we have some use that is already approved. There is a plasma machine that is approved for medical use in Europe. That's the one I have in the lab, but not in the US. And I believe it's not approved in Brazil as well. So plasma, again, it has antimicrobial properties because of some of the products that we have around the flame. And it also has some regenerative properties. For example, one of my colleagues that I work with in plasma research, Dr. Coelho from NYU, he is looking into osseointegration, and plasma is able to increase osseointegration in implants in animals. So we can combine them, anti-biofilm effect, mostly killing the cells that are inside the biofilm, and an osseointegration effect, which would be excellent. So I've been studying plasma for a few different possibilities. One of them is we looked into plasma in a dual species biofilm that was Candida and S. aureus, looking into a possibility for decontamination of prosthetics, for example. So we looked at this plasma in this pathogenic oral biofilm that is known. It's made in the lab, so we can control it. It's not as complex, and it's far from mimicking what is happening in the mouth, but it's a controlled study where we can start, right? And we also looked at how this treatment would affect the reconstituted oral epithelium, looking more towards toxicity, right? So we had a biofilm where we used two species, Candida albicans and S. aureus. Why these two species? Candida, as I mentioned, it's in the mouth. If we have prosthetics, that might grow in that surface, and then we usually have mucositis, and that is very difficult for the patient to deal with. So if we could either clean the prosthetics or treat that mucositis, that would be very helpful. S. aureus usually binds to Candida, and when Candida usually forms hyphae, and when they form hyphae, the S. aureus binds to it, and the hyphae penetrates the tissue. So S. aureus has a way of entering the system, causing a systemic infection, and that is actually very problematic for patients. So we had a biofilm model, which was designed according to our research questions. So I have resin discs where the biofilm grew on, same resin that we use to make prosthetics. And we grill them until they reach a mature stage for those two different types of cells in that specific design. And we tested, we treated this mature biofilm with plasma, one signal treatment. We want something that can kill Candida, which is an eukaryotic cell, in the form of biofilm, which is its pathogenic form. So we also wanted to know if this dose would also be toxic for the oral epithelium. If we have a yes here, that is something that unfortunately we could in the future even think about using in a patient. To do that, we use this reconstituted oral epithelium. It is tissue, just like the human tissue, but it's made in the lab. So it is as complex as the human tissue. It is very controlled. So we know everything that is happening there, the age and everything. So it's easier for us to compare because the parameters most of them are already known. So we use this tissue, we buy it from a lab, it comes to our lab, and we have 48 hours to use it. So we treat this tissue with the same amount of plasma, which here is usually regulated by time and distance from the tip of the equipment. And then we did several tests in this tissue to have an idea about if it is cytotoxic to the tissue. We saw that it did have an antimicrobial effect when this biofilm is actually established. So we saw a decrease in recovered cells when we compare to the control. When this we do just by culture, very simple. It does give us information, but the only thing we are seeing there is how much cells we can recover, and we compare it with our control group, right? So to take a deeper look, we did a confocal microscopy where we have Candida in the first column. We have Staphylococcus aureus here, and we have a combination of the two in this third column. So we can see that when we treat with plasma, we have a lot more dead cells. We can't see dead cells when we culture, right? We only see the whatever we are recovering. So it's nice here that we can see the dead cells. We used drugs that are gold standard as positive controls for them. Then we did scanning electron microscopy, where we can see the cells, right? So we can see all this different, what really happened in the cells. And we can see that in the hyphae here, negative control, that's very normal. And then when we go to the plasma, it was actually doing a real mass, right? If we look at, see that I mentioned that S. aureus binds to the hyphae. So this enables it to penetrate the human body. Plasma also made a mass here. Again, as I mentioned, okay, it does kill Candida, S. aureus when they are in biofilm, but is it also toxic to the tissue? And no, it's not. Not in this dose. So if we look at the negative control and plasma here, cell proliferation is very similar and different from the positive control, and cell apoptosis is also the same for the negative control. So that is a good sign. Our cells are much better to protect themselves than Candida, which we already knew, but of course we have to show that. Another use for LTP would be for periimplantitis. As I mentioned, we know that there is osseointegration when we treat with plasma, and we know that the periimplantitis, so patient put an implant, within the time it gets contaminated, and then usually this implant will be lost. We have to remove it. The idea here is to try to save that implant. So during the surgical procedure, we open a flap, we would decontaminate the implant, and we would close that flap and then see what happens. So we started by just looking at a simple single species biofilm of Porphyromonas gingivalis. It was a 5-day-old biofilm, which is already mature. And then we looked at recovered cells, which is in here. So we had a decrease in the log of CFU per ml. So 2 logs decrease here when compared to the control, about 1.5. And then when we look at dead cells, we see dead cells here. We actually saw a lot less biofilm here than in the negative control. It was a single species biofilm. Everything in there is helping the biofilm to grow. When we looked at tissue here, but not our epithelium anymore, it's the same reconstituted tissue, but here is gingival epithelium. We see very similar, it's not toxic to the gingival tissue. If we look here, this is 1 minute of plasma, 3 minutes, and this is the negative control. While when we look at the positive control, it's really kind of destroying it. So let's go further and look at a more complex biofilm. So now we have a model where we know the bacteria we are dealing with. We know, we tested this model to see if that after 24 days of growing these 4 bacteria together in these conditions with this media, we did fish, which is fluorescence in situ hybridization, and we were able to see the 4 different species there after 24 days of growth. So you really have a multi-species biofilm until the end. So those are the 4 bacteria we used. They're present in periimplantitis, so that's why we chose them. And now, this is not published yet, by the way. We are just submitting it for publication. So you guys are probably the first ones to see this data. But we looked at plasma treatment, and we had several other controls because we have several different types of bacteria. So we have to guarantee that we have a control that would work in each one of them. And also, we use different time and distance for plasma application. We did 24 hours, 3 days, and 7 days biofilm is what I'm presenting here. We are now working on older biofilms, and that's what we see. So if we look at the box plot results, we have here 24-hour, 3-day, 7-day for each one of the bacteria, but they grew together. Okay, we just counted them. So what we see, we weren't able to recover almost any one of them with 1 minute, 3 millimeters. If we make it the distance a little bigger, 10 millimeters from the surface to the plasma tip, we need 5 minutes of application to be able to completely kill. So that shows that whatever is in there is different, right? So 1 minute would be a very short application. So that would definitely work in the clinic. So now the next step, after we are done with that, we will go and look into animal studies. This 1 minute, 3 millimeters, we can see all the that cells right after the plasma application. So for the 4 species, it's working. Then, so I'm talking about treating biofilms, right? So we are getting biofilms that were already formed and treating them. Can we prevent this biofilm from growth or maturation? So that is another study that I have here that I show that we can actually test things that would prevent. Again, we have to, the question is, if I have a situation like dental caries, where you have an inoculum there in the mouth all day, every day, it doesn't really matter if I treat that biofilm once, right? I have to keep treating it. So we looked at blue light, white light. So of course I had other tips, right, to look at blue light, but light, we have light and our organisms have some sort of sensory system to detect light. Our body, we use light. There are hormones that only work in the dark. Vitamin D, we need the sun, right? That's the same for every other living organism in this planet. Bacteria use light, plants use light, right? This is my daughter, when she was in 3rd grade, she was doing a science project to see how energy transfers, right? So even the energy that we have, the energy I'm using here to talk to you guys, it comes from light. The sun transfers energy to the corn, that transfers energy to the corn chip. So it's processed, corn processed, all this energy is being transferred. We eat the corn chip and then we have energy to jump. So that is as simple as it gets. It's showing how our organisms can be influenced by light. Now she's 15. This is her. She's looking into other aspects of light, blue light here. But I like that because it's a simple way for us to understand how light really interferes in our daily lives. And same way for microorganisms. So here I studied the effect of twice daily blue light application in the development of a matrix-rich biofilm. I use an S mutans biofilm here because it does form a matrix-rich biofilm if I give sucrose to it. So that was the model we had. And when we look, blue light, it had some antimicrobial effect. So the recovered cells here were less than the negative control, which was saline, but not as good as chlorhexidine. Chlorhexidine is a really good antibiotic, right? It really kills the cells. Dry weight was similar. But then if I measure the amount, we measured here the amount of EPS insoluble in the matrix. And if I normalize it by dry weight, which means that it's not related to the amount of cells that are there. So if we have less cells, less production, so we have lower. No, I normalized it. So each cell, it's almost like looking at what each cell is producing. We can see that blue light had very little production of extracellular polysaccharide. We can actually see that when we look at the confocal. If we look at here on this line number 3, this is a very thin biofilm, even when I compare with chlorhexidine. Another interesting fact here is that we can see that light, whatever is killing chlorhexidine, it's killing on the top of the biofilm. It's not really being able to penetrate, right? But this is just another thing that we saw. But that's actually very neat. I can use light and decrease the polysaccharide formation, right? Another thing that I want to talk about very quickly now, I'm almost done, is that, as I mentioned, we have to compare, we need to have our question and then choose our assays, right? So it's really important to understand that depending on what you're studying, the assay has to really be well designed. So I did here, a lot of times we do imaging and we do regular scanning electron microscope, but we need to fix the sample, right? We have a fixation process of the sample before we can look at the sample under the microscope. So one of my students did once a project where she compared the SEM and what another instrument that we call variable pressure SEM. By the way, the UFC has a great equipment that does this sort of images, which is quite rare. It's not as accessible as the regular SEM. And then we were able to see that the SEM here, probably because of this whole process of preparing the sample, the matrix is completely dried. I have a picture here that shows a little closer. So the matrix is dried. We can see the cells, right? But the matrix is completely dried. When we look at fresh samples, though, which we can do with this equipment, we can see that the matrix is a lot more preserved. Here a little closer. So you can see here the S mutant cells, and the matrix here is completely dried. So for a long time, all the pictures we saw, and actually we still see, are SEM pictures. They're fine to look at the cells, but they're not good to look at matrix. So if we are looking at studying matrix, we really need a better way to visualize it. So the variable pressure would be the equipment of choice. Physical properties is another thing that we can look at. I met with a professor from the physics department once, and we were talking about looking at physical properties. This is holographic microreology. It's a technique they use to study viscoelasticity of polymers. So we decided to try that and look at viscoelasticity of polysaccharides, which are like a gel, right? So this technique uses microbeads that are glass, and then they use a camera to look at how these beads move in a certain frequency. So they move the slide, and then these silica beads that are inside this gel, which in this case was polysaccharide, they will shake it and it moves. Then they trace the move. So if we look at polysaccharides that are extracted from the biofilm, and each one of them, this red balls are the silica beads, they're the same size. I just had to make this one bigger so you could see that it's actually moving. But if we look at water-soluble polysaccharide, we can see that it's a lot more flexible, right? So that means it's a lot more elastic. And the insoluble is a lot more viscous. The most viscous thing is solid. The most elastic thing is liquid, right? So that is just confirming what we already knew, but maybe we can use these techniques to look at viscoelasticity of polysaccharides and oral biofilms. Here, for example, we see in the chart D, increasing the frequency that we move the slide, we have a decrease in the viscosity. That's exactly what happens when we brush our teeth. We are increasing the frequency that this gel is moving, and then we decrease viscosity, making it more elastic so we can disrupt this biofilm. If we can find something chemical that can do it, that would be interesting. So I'm just going to leave this open here for you guys, some challenges that I find by looking into biofilm research, and we can you can put your mind into it and try to discuss and think about. First thing, I always try to understand the complexity of a healthy biofilm. A lot of times we focus on disease and not really health, but understanding what is a healthy biofilm is really important so we can actually look at disease, but understanding that this is like just an imbalance. So try to bring the environment back to balance is definitely the key. So we can either create conditions that are more linked to health, the oral health, let's create conditions where we can have a healthy biofilm there. Sucrose being probably the worst, our diet, right? Understand that assay, depending on the assay we use, we always will have good things and bad things. So it's important that we know the what is actually what we are looking at when we choose an assay. And of course, to always try to follow the steps to implement this biofilm research into the clinical practice. That is actually the ultimate goal, right? That of all this research we do. So with that, I think I took a little longer. I'm sorry. I get very excited. But with that, this is, I finished my presentation. I want to thank you guys again. And let's hear from the other professors, and then we can discuss it a little better in the end.
So Professor Simone, congratulations for your brilliant presentation. As we said before, and you just confirmed, we are going to discuss after all the presentations. So congratulations again, and thanks again for collaborating with us in the graduate program at UFC.
Oh, it's my pleasure. Thank you.
You are very welcome. So now we are going to the presentation of Professor Luiz Bertassoni. So again, Professor Luiz Bertassoni is from the Oregon Health and Science University School of Dentistry in the United States. I would like to say some words. I think that the first time we met, we were in the Penn Perio meeting in Philadelphia. I think it's 2017, and I think when I saw his presentation, I thought, look, this is something that we used to watch in the movies when we were kids, so probably it's very exciting to work with the 3D printing, bioprinting, whatever. And thank you for collaborating with our graduate program too. So the presentation of Professor Luiz Bertassoni, it's called Machine-Made Human Organs on the Frontier of 3D Bioprinting. So Professor Luiz Bertassoni, please.
Thank you so much, Rodrigo. Thank you, everybody at UFC for the invitation once again and for the collaboration. I'll bring my slides up in a second. It's always a pleasure to be with you guys, even though I can't be there, unfortunately. But as you know, it's always a pleasure to be around. I always had a great time every time I visited Fortaleza, and hopefully we'll be together very soon. So, Rodrigo, if you could just confirm to me that you can see my slides.
Not yet.
Okay. Let me know when it gets there then.
Okay.
I can already see it in my screen here. Let's see if there's anything.
There you go.
Okay. Perfect. All right. So I took this opportunity to try and summarize some of the things that we've been involved with in this field of bioprinting and in other methods of engineering human organs and organoids and tumoroids and kind of like structures that resemble tissues in the body, and not only sort of like what we've been doing specifically in our lab, but since we had a broader audience, I figured I should take you guys onto a chronological history telling of how we got there and sort of like where we are so far and what are the things that are going to happen in the next few years. So it doesn't cover only my research, but it covers some other exciting things happening in the field. So the need for this idea of machine-made human organs is very high. And the reason for that is because only in the US, there is about 120,000 people that are waiting for an organ transplant. So it's one of the biggest problems in medicine, the fact that you literally have to wait for somebody to die so you can actually cure a patient. It's not particularly convenient that you have to depend on death for that. So it's interesting that, there is 22 people every year only in US that because of this shortage, they end up dying just because there isn't enough organs for transplants. And what is more interesting is that even though 95% of the population supports this idea of organ donation, only 51% is actually signed as a donor. So you can understand the shortage there very easily. So bioprinting essentially started from this idea that we can use engineering methods to address this shortage and start engineering organs that can, be addressing this need in the field. And this is a drawing that was made for Revista Saúde there in Brazil that we gave them some interviews and some discussions of our work, and they built this nice drawing of what the sort of like the concept is all about. But I'll get you to the step-by-step on how we got there. And first, I think it's interesting to mention how we even started, right? So I did my postdoc when I started doing tissue engineering work at Harvard and MIT, and there was this rumor that went around in the labs that we worked that the field of tissue engineering started with these two doctors, Charles and Joseph Vacanti, and Robert Langer. He's an engineer. So these are two physicians from Harvard Medical School, and they were friends with Robert Langer, who was an engineer at MIT. And I did part of my postdoc in his lab, did lots of experiments in his lab, even though he wasn't my direct supervisor. And the rumor goes that these guys essentially got together at the end of the day and went to have a beer, regularly. They were friends and they were just talking to one another. And then the doctors, in talking to Bob Langer, Bob was asking, what is the biggest challenge that you have in medicine? And then they said that, is the fact that we don't have enough organs for people that need them in order to save their lives. So the engineer talking to the doctors, they basically had this crazy idea after several beers that, well, why don't we just go ahead and engineer organs? And it sounded like a crazy idea at the time because literally there was nobody that was trying anything like that. And then a few years go by, and this is what happened, and you probably remember this.
1997, Boston, Massachusetts. At first glance, it seems impossible. Yet like something out of science fiction, this animal may actually become a trailblazing hero. American scientists have successfully grown and attached a human ear onto the back of a mouse.
So you all probably remember the news when this came out, right? Like the ear in the back of the mouse. And this was essentially a result of a happy hour probably too many drinks at a bar with an engineer, with a couple of physicians having these crazy ideas, and that's how you begin to change the world. So how do we even get there, right? It all starts with stem cells, and that was essentially the first driving factor for a lot of these findings. And I brought a quick sort of like illustration here that I hope you guys are hearing from the video that explain what the stem cells are in a very pedagogical way. So I'll let it speak for itself.
Stem cells are cells that are undifferentiated, meaning they do not have a specific job or function. While skin cells protect your body, muscle cells contract, and nerve cells send signals, stem cells do not have any specific structures or functions. Stem cells do have the potential to become all other kinds of cells in your body. Your body uses stem cells to replace worn-out cells when they die. For example, you completely replace the lining of your intestines every 4 days. Stem cells beneath the lining of your intestines replace these cells as they wear out. Scientists hope that stem cells could be used to create a very special kind of personalized medicine, in which we could replace your own body parts with, well, your own body parts.
So there you go. That's the concept of stem cells, right? So just this ability of actually getting cells from your own body to sort of like create different types of structures that you need. But what's interesting about cells is that, as you're seeing here, cells interact with their surroundings a lot. They just move around, they recognize one another, they know where they have to go. Every now and again, you're going to have like a crazy cell here that doesn't really know what to do. But by and large, cells are pretty good at interacting with different surfaces. And once they interact with these surfaces, then they begin to divide, they begin to proliferate, they begin to express different markers. And if you're able to control the material that the cells are interfacing with, then presumably you should be able to facilitate some of these processes. You should be able to facilitate cell division. You should be able to promote cells moving around in different locations. And that's essentially the gist of the field of tissue engineering, is that you're going from cells that are responsive to surfaces and you're coming up with engineered tissues altogether. So way back then, at the time when Bob Langer and Joe Vacani came up with this idea, the concept was that you would take stem cells from the bone marrow from a patient, just like as you see in this drawing here. You would isolate these cells, and then you use a combination of these 3 different things: growth factors, a biomaterial, scaffold, and the cells that you just isolated. And then you would seed the cells on top of these biomaterials after these biomaterials are built, and you put these growth factors or cytokines to stimulate cells to do things. Now, you fast forward 25 years or more, and what we have right now is that we don't even need any of that anymore. So we don't need to rely on stem cells necessarily anymore. We're pretty good at coordinating the interactions of fully differentiated cells. And even more importantly, you can get cells from anywhere in the body, such as your skin or surface cells that are readily available, and you can transform these cells into these induced pluripotent stem cells and then get them to differentiate. So there's not such a high reliance on stem cells isolated from patients anymore. And then not only we can take these cells and just cover a structure that has been printed, or seed these cells on top of something that was created, but now we can actually do things at the same time. We can create what we call these 3-dimensional cell-laden biomaterials. We can literally embed or encapsulate cells within these materials and then biofabricate these materials with the living cells in the process. And that's where the field of bioprinting comes from. So you can play with these materials, you can get them out of these syringe needles, out of bioprinters. We call them bioinks. We can do all sorts of different things. What are these materials? The majority of these materials where you can encapsulate cells in 3D are called hydrogels. And then these hydrogels are very good for cell manipulation and for 3D bioprinting for a number of reasons. First, because just like the human body, they have a lot of water. They can be made out of several different types of polymers. They can be protein polymers. They can be synthetic polymers. They have a highly controllable structure and physical properties. And they can also be loaded with different types of solutions, right? Growth factors, cytokines, cells, other types of cues, as we named it. So for instance, things that we have done in our lab, we can take tissues in the body, isolate the proteins, such as, proteins found in the tooth or in bones or skin or even the heart. And we can transform these proteins into this clear hydrogel material, which basically allows us to just get cells, mix them up with these solutions, and then we end up with this three-dimensional cell-loaded biomaterial. And then by functionalizing these with methacrylates or things that we, for instance, have used in dentistry for several years, we can use things such as a dental curing light to set these hydrogels, to photocross-link these hydrogels, and then create different types of shapes and structures. And we can play with that chemistry quite a bit. So a few examples of, how we started using these machine-made methods to control tissue structures can be the example that I have here, where we use computer microchip fabrication technologies to actually make human tissues. So everything that we use that is electronic nowadays, your cell phone, your computers, your TVs, everything that has a microchip device inside uses a method that goes around this idea of lithography. So this is one type of lithography. It's a photolithography. And basically what happens here is that you have a light source, and then you create a photomask, which is very, very, very small. And then you have a photoresist under this photomask. And basically what the mask is doing here is creating a micro pattern on that surface, and then you convert this photoresist into a silicon wafer, and that's how you end up with these, these different lines that you're seeing here in this microchip. These are all made by lithography. So some people that are far smarter than me way back then started playing with these methods to actually make tissues. So you could do these instead of using a silicon wafer. You can do it with those hydrogels that I showed you earlier. So you would put gels on a flat surface, you create a photomask, you put that photomask on top, and then you expose that hydrogel through a mask to either UV light or a dental curing light. The cells tend to tolerate that quite well if you do the chemistry right. And then you end up creating structures like that. So this is a logo for our university. We did that project a few years ago. We put some dental cells on it, and we saw that they aligned fairly well. This is some work that I helped with when I was at Harvard, and these are cardiac cells. And you can see that if you put these cells on top of these pattern materials, they actually beat synchronously. And if you do it and put it in water, you can actually see the, a structure of a beating heart cell that is induced by this micro patterning in solution. So all fine, all good, very interesting stuff, but humans are not flat like you saw before, right? Even though we can create these very intricate flat structures, we know that we're built in 3 dimensions. And that's how the idea of 3D bioprinting started taking a lot of traction in the field. Because you can take some of those technologies that we've used in the past, and now you can bring a 3rd dimension to it and start creating things that are more resembling of the structures in the actual body. So where did we start with the 3D bioprinting? Well, if you go back in history, you find that the first thing that was done was to take a regular cartridge ink from a conventional HP printer. There was another person that was very curious and said, well, let me empty this ink from that cartridge and let me put cells inside. Let's see what happens. And sure enough, they were actually able to dispense cells out of a conventional office printer, and they were able to put cells in a piece of paper. Obviously, at that time, the cells were all dead and they didn't tolerate the process because this thing was not designed to actually dispense the cells. But what you're seeing there is basically the jetting process of an ink, which could be performed either with cells or with other types of materials, and even paint, which is what we use for actual printing. So you take it from your two-dimensional cell printing, and then you put actual live cells, which is what people are doing nowadays. And then what you can do is start replicating, some of the complexity of very intricate paintings, for instance. But now this is all done with live cells. And the reason why you don't see the exact same image here is because these cells are alive. So once you print them, they just keep moving, and basically they change the painting, and it's literally a living painting that you're seeing. So what these guys did was essentially they tagged cells with different colors, right? Primary colors. So they just tagged cells with 3 colors, red, green, and blue. And basically by the combination of the number of cells that you get, you can create the different colors here. And then basically they recreated this painting just by printing this through an inkjet printer, which is something that we also have done in our lab. So all interesting, and it gives you the ability of actually, using household tools such as a conventional inkjet printer to print cells. And there's even papers that you can find nowadays, such as this paper on this journal, Jovi, that actually shows you a demonstration of the step-by-step of taking your HP printer from your office and turning that into a cell printing machine, which is basically anybody can do it nowadays. So very interesting stuff. But again, this is all in 2D. And how do we go from 2D to 3D? And that's how that process begins. 3D depends on our ability of actually inspecting and understanding tissues in the body in 3 dimensions. So this can be done with an MRI or a micro CT type of system. You generate a DICOM file, you take that DICOM file, and then you create a digital reconstruction or a 3-dimensional rendering of that image that was collected. And then out of that, you would generate a computer-aided design file, basically to create the part that would sort of like fit that missing piece there. And then one thing that is critical for bioprinting is to ensure that you can, as we call it, slice that 3-dimensional structure into different layers. 3D bioprinting stems from the ability of being able to create tissues one layer at a time, and subsequently sort of like just add layer on top of one another. And that's where the idea of additive prototyping comes from. So the way that this works is you take those, very frequently, you take those hydrogels that I showed earlier, you embed the cells within these hydrogels, and then basically you would go around replicating this layer by layer design with the cells already embedded in the system. And that's an example of what we're doing here. So this was a bioink that we developed out of a piece of a tooth. Actually, we created a hydrogel that had dental proteins. We embedded those hydrogels with living cells, and then we could go around and create all sorts of different intricate structures, and we're able to show that those cells remain alive through that process. They tolerate the process really well. We could create structures such as, writing the name of our university or other things like that. And it's all very straightforward for us to do. And in fact, we're excited to start with that at UFC very soon. Hopefully, we know that you guys have acquired a printer that can do quite a bit of that, and we are already working with the team to get the ball rolling. So I'll give you some examples of what we have actually accomplished with these different types of printing. So the first thing that we wanted to print was the vasculature. The reason for that is because the vasculature is basically the plumbing in our bodies, right? All of the pipes that connect everything. Without oxygen, cells essentially die. So the vasculature needs to be very close to every cell in the body to provide oxygen and nutrients, and also to get rid of all of the bad stuff that we eat on the weekends, and right, the beers that we drink and all of that stuff. So it's all controlled through the vasculature. So the method that we developed back in 2014 to bioprint the vasculature was fairly straightforward. We found a type of material that didn't adhere to a hydrogel that was loaded with cells. So we would print this material as a template. We would cover that material with a cell-laden hydrogel, and then we would sacrifice that material and end up with hollow channels in the middle. So this is a photograph of that 3D printed template in green, and the blue around is basically the cell-laden hydrogel. You can see that this sort of like bifurcates in 3 dimensions. And after we sacrifice that template, the printed template, you can see these microchannels inside. And that's how the sacrifice process looked like. So you would aspirate that and they would sort of like come out. We could make structures that were as small as 150 microns, which is pretty good. And then this is after we put it in the incubator for 14 days after we load endothelial cells inside of these channels. What you see in white here, these little structures, these are actual living blood vessels that were made in the body in a question of 2 weeks. How do we know that? Well, those cells, they should fluoresce under the UV light because they are GFP expressing. There are proteins there that should autofluoresce under the light. And sure enough, once we put it under the light, you can see the green there. So that is a confirmation that our 3D printed blood vessels are there. When we put these under the microscope, you can see that they have the same structure as the vessels in the body. So this is a monolayer of endothelial cells. It's a nice and long tube. These cells, they can sprout into the matrix as the cells in our bodies. And as you can see here, right, like this is showing sort of like the entry point of one of those vessels, and the vasculature is clearly forming there. We could also take this vasculature or these capillaries and dissect them just like you would do in your anatomy class. And you could even hold and pull these blood vessels by hand. They were empty, they were hollow. You could flow stuff through them, right, as you would expect from a blood vessel. And now we're even able to put different layers of cells around these blood vessels. So this is more and more consistent with the microstructures of the vasculature in the body. And we have recently used this model to give you one example of many that we have used these printed vessels for. We were recently able to detect the extravasation of the SARS-CoV-2 virus through that engineered vasculature. And we haven't published this yet, but we're in the process of. We ended up finding that depending on the levels of inflammation that we know is a problem for COVID-19, we can basically, inflammation facilitates the extravasation of the virus through the engineered vessels and the vessels in the body. And those, the viral particles, they tend to attach to perivascular cells, which are present everywhere in the body. And we believe that this is one of the reasons why there's so much compromise in COVID-19 patients, because the virus is actually working around the vasculature and not necessarily only on epithelial cells. So this is just one example. And when we published that, that was a big hit in the news. It was covered by a lot of different news cycles, and it was selected as one of the top 100 stories of the year. We were even had a big effect in the stocks market. So there was a big boom in the price of the stocks exchange market for biotechnology. And we're very excited about that. So we obviously continue to work around that. Another way that we print blood vessels is using a different type of bioprinting. In this case, it's not an extrusion printing, it's a light-controlled printing. So the way that this works is we fill a vat with hydrogels loaded with cells, and then we basically create these tiny little microparticles of hydrogels, such as the ones that you're seeing here. These are about 250 microns to 500 microns or more. And then this is more or less what the process looks like. We can print these microparticles very, very fast. So this takes about, I don't know, like 5 minutes to print. I'll speed this up. So you can see that we're printing structures there one at a time. And every spot that you see in that video there, it's a quadruplicate. So we call these microarrays. We can put cells inside of that. Sorry. And if we load this with endothelial cells, we know that these cells will form microcapillaries. And this is what you're seeing here. Now, because these cells are, or these microparticles are so small, we can inject them through a syringe needle. And what is exciting about this is that we can effectively engineer blood vessels in the lab, grow them to the point that they're very happy, and then we can take a syringe and literally squeeze these blood vessels into somebody's body minimally invasively. And what these images here show is that these vessels will go on to anastomose with the host vasculature and recreate the vasculature in a site that has lost it. So we're using this to regenerate the tooth. We've tested these in the brain. We're looking at heart regeneration and a number of other things. And we know that this is a very effective way of regenerating tissues in the body. So the other tissue that we're interested in printing is the liver. The liver is the site for most problems in pharmaceutical companies. Basically, everything that you try in terms of drug development, it tends to fail in the liver. There's a lot of toxicity. So this graph here shows basically the number of potential drugs in medicine and how clinical trials essentially go down and narrow it down to essentially from hundreds and hundreds of possibilities of new drugs to only one that works, and a lot of the problems are in the liver. So our idea was if we could bioprint the liver and test drug toxicity, that would be great. That was the first project that I started when I was starting with bioprinting here in US, and this is the printer that we used. It's an Organovo bioprinter. And the way that we developed this method was to aspirate cells into this capillary here. So basically, again, you have a cell-laden hydrogel. This is photopolymerized. This was the first time that somebody was able to bioprint these photocross-linkable hydrogels. And this is what the printing would look like, right? So the fiber is getting extruded out of a rod, and then essentially the positioning of these fibers allows you to create different types of three-dimensional structures. So this was almost 10 years ago, and now the field has evolved considerably, but it was the first time that we're able to put, these liver cells in a photocross-linkable gel, keep them alive, which is what you're seeing here. The cells are kind of like expressing the markers that they should. We could stack up multiple layers, create these tiny little holes here to facilitate gas exchange, oxygen, and nutrients. And basically, we're able to show that we could keep cells proliferating, we could keep them alive. They would secrete albumin, which is a marker for liver physiological response. And then we would bioprint these cell-laden hydrogels inside of these micro devices and expose them to drugs. And then the team continued this work later, and they were able to find that you could replicate the toxicity of these anti-inflammatory drugs here, very closely matching what happens in the body with these bioprinted tissue constructs. So this is something that is been evolving. Another thing that has happened in the field very recently is this idea of bioprinting full beating hearts. And this is quite exciting, in my opinion. So there was this team from Carnegie Mellon University in Pittsburgh, and they developed this method where you can bioprint or 3D print collagen within a bath of microspheres. These microspheres are degradable. And what is interesting about this is that the microspheres are essentially holding the fiber that is being printed in place. So it's literally like this is floating, but the microparticles are robust and rigid enough that they actually hold it in place. And then after you print, then you can just dissolve these microparticles and you end up with the tissue. So that's what that looks like. So they're printing inside of this jello-like material. And then after you dissolve the jello-like material, you end up with your printed structure alone. So they use this method, which is very fancy, to bioprint a full-size neonatal, so it's a pediatric full heart. And here they are showing kind of like the different structures of the heart and how they were able to fill in different cavities and different things. And this video here shows more or less what that process looked like. They went ahead, took a micro CT of the actual heart, and then they went on and replicated that with that bioprinted structure. I'll speed this up so we don't get caught up. And then you can see all of the kind of like inlets and outlets of the vasculature. You can see the different arteries and veins. You can see the different chambers. So atrium, ventricle, and the valves, and basically everything that should be there is there. So all of the structure is essentially replicating the ones in the body. And then when they did a more simplified structure that is actually filled with cells, then sure enough, that's what you get. A fully beating heart that has been bioprinted, and it has pretty controllable beating patterns, just like the heart in humans. Obviously, the microstructure of these guys is not as complex as that, but I'm pretty sure that that's where these guys are going to with these next, and this is an exciting time in our field, most definitely. So another focus of this regeneration has been cartilage and bone for craniofacial applications. This is particularly relevant. And there has been some interesting developments on that too. So this is another work that was not done in our lab. This was done by Anthony Atala's lab. He's a collaborator of ours and has been a pioneer in the field. So these guys use a slightly different system. It's a bioprinter that they've developed. It has 4 different inks potentially. And basically what they had here is a combination of methods that have been used in the field for different things. So they picked up some of the things that we've developed. They picked up things that other groups had developed. They developed their own and combined this sort of like hybrid method. So the example here, they printed a supporting structure in the PCL, 2 different types of cells, and a microchannel using that sacrificial method that I showed earlier. So that sacrificial material was pluronic, and basically that can be sort of like diluted instead of aspirated, and it creates something that kind of looks like what the vasculature would be doing without the endothelial cells. So this is the printing going on, and the process that they use is similar to what we mentioned earlier. You take an image of the MRI, then you generate a CAD model, and then this is a visualization of the lines that would be printed. It actually shows the pathway of what each line would be doing, and that's what you're seeing there. So you print a layer of the supporting structure, and then you come with the cells, and then the printing process takes place, and then what you end up with at the very end of the printing process is something like this, right? So this is a bioprinted tissue product, and it doesn't quite look like an actual human organ. What is interesting here is that, again, this is the size of a human ear, a pediatric human ear. So we're talking about an actual structure that has the size and the cells in the body. And even though this doesn't look like a human ear right off the printing, have a look at this. After you dissect and you implant this and you let it grow, this is after you remove basically the skin from around the ear. And you compare the histology, the human and the 3D bioprinted, there's virtually no difference whatsoever. And that's what they showed in this paper, that you are able to replicate both the microstructure, the biology, the mechanical properties, everything that you need is there. And it's very hard to tell them apart. They're basically the exact same thing. So in terms of bone, our lab has developed methods to regenerate the bone with the complexity that it has in the body. So we developed this technique that we can encapsulate cells in a hydrogel, and then we basically calcify the cells in place, and we replicate the complexity of the nanoscale mineralization that happens in the body. This is not relevant for the time being. And we can create these in any type of structure, shape, size, or anything. So what you see here is essentially the gel with the cells before the calcification. And in a matter of 12 hours, we can get this to calcify, and we can essentially make bone in any shape or size. And this is what you're looking at. So this is something like a TEM image, and we're looking at this layer by layer. So you're seeing cells, and the black dots are the minerals, the gray dots are collagen fibrils. And if we now digitally remove the collagen, you see how these cells are fully embedded basically in a rock of minerals. And this was the first time that anybody was able to show that you can basically cement cells within minerals. Previously, it was thought that this was impossible to be done because cells wouldn't tolerate, they would die, they wouldn't have enough oxygen, there would be too much osmotic pressure around the cells. But it turns out that we were able to show that you can embed cells within minerals, and it turns out that they actually like it a lot. So when we compare the microstructure of our engineered bone on the right against the human bone, it's virtually the same. And we can embed both bone cells, we can embed blood vessels, we can embed nerves, we can basically recreate the whole thing. And that was the first time that anybody was able to also engineer bone with the vasculature and the innervation. We published that recently in Nature Communications. And what is interesting about this is that not only we can use that for understanding biology, but we're finding that this is actually better than what is out there in the clinic for bone regeneration. We don't use any growth factors, we don't use anything. This is basically just the engineered bone. And when you compare this with BMP2, which is something that has created a lot of problems in the clinic actually, but it's still the only solution when you can't have access to autologous bone, we find that our engineered bone creates more bone volume, more bridging area, and the quality of that bone is also comparable to the BMP. So we're excited about that regeneration. So where are we going now with these engineering methods? This is something that we're doing right now in our lab. If you look at a complex organ, and I use the liver here as an example, right? It's a very, very, very intricate three-dimensional structure. The liver is composed of individual lobules. And if you look at it, literally every cell that is here is doing something different. So it is extremely important to be able to replicate the specificity of where each cell should sit in a three-dimensional space. So we're working with this company from Sweden, where they developed this method where we can bioprint one cell at a time. We can create structures that have complexities down to the single cell level. This uses a microfluidics device similar to what I'm showing here. It doesn't really, I think, matters exactly the process, but it's a microfluidic system that allows cells to be dispensed one at a time, and for us to switch from different cell types in the middle of the print. So we don't have to stop, go back, get another cell, put it on. You can literally just keep switching cells as you're printing. And then you're able to print structures such as, like these more complex, very cell-rich regions here. This is just 200 micrometers. You can literally see where each cell sits. You can create things like the liver. You can create things like the skin cancer model. Obviously, this is not intending to replicate that yet, but this is what we're working in the lab. This is just a proof of principle. But what is interesting about this method is that we can even print one cell. This is what you're seeing. So it's one cell separated by one another by about 400 microns or so, or less, maybe 200 microns. And you can literally put one cell at a time at the right place at the right time. We can make clusters of cells, or we can make lines of cells. And this video here shows us printing. I'm going to speed this up again. Printing, again, the name of our university with these cells. This is fairly quick. We can do that, in a matter of 5 to 10 minutes. It's a little bit hard to see with the print head on top, but I'm going to show after the printing is done. It's probably going to be a little bit easier. So there you go. So this is what we just printed in a matter of minutes. So you can literally see one cell at a time. And at the end there, we reduce the line thickness, and you can see here that we can have a line of, a single or two cells side by side. So we can start replicating some of these structures here. We got this machine very recently. That's what we're doing now. The last thing that I want to talk about is this idea of machine-made organs that are miniaturized. So this is a field that's called organs on a chip. And the idea here is that we're not trying to regenerate anything. all that we're trying to do is to recreate the microstructural complexity of human organs in a micro device that we can keep under the microscope and essentially watch as biology happens before our eyes. So there has been multiple examples of organs on a chip. You have brain, heart on a chip. The first one was the lung on a chip, the kidney. We have published on a number of these. And I think it's fair to say that we are leading the way in terms of calcified organs on the chip. We have a model of the bone. We have developed the first model of the tooth. We have a company to engineer bone marrow on the chip. And there's a lot of stuff happening around that interface. And I'll give you one example of that. So one of the things that we're interested in understanding is the process of cancer metastasis into bone. The cancer metastasis happens through this interesting phenomenon where cancer cells extravasate into the vasculature or intravasate into the vasculature. They travel through those capillaries. They are attracted to the bone site because of a lot of different cytokines that the bone cells secrete. They squeeze through the vasculature and then they basically lodge into the bone tissue. Very little is known about that specific process, why cells go there and not elsewhere, why they become more aggressive once they metastasize, all of these things. So you saw earlier that we engineered that method to, or developed that method to engineer vascularized and innervated bone. And because we have a lot of knowledge on these organs on the chip field, we figured that we could miniaturize and create essentially a machine-made vascularized bone that would interface with a cancer type, in this case, prostate cancer, so we can understand that process a little better. So what we did here, we developed this chip. It has a piece of bone in the middle, and we would load side channels with prostate cancer cells. And what you're seeing here is basically the engineered vasculature inside of that miniaturized bone in this microchip. The reason why we engineered the vasculature is because we want to watch as the cancer cells travel through these blood capillaries and eventually extravasate into the engineered bone, so we can understand that process of metastasis a little better. And what you're seeing here is basically the vasculature in green, the cancer in red, and I'll draw your attention to these circles and to these arrows. So if you look at this one in the middle here, you can see how the cell is traveling through and then eventually it extravasates. So literally we're replicating the process of cancer metastasis into bone in a microfluidic device. We're watching it as it happens. We can take these apart. We can do a lot of biology on it. And we believe that we have recently found an unknown mechanism by which the process of cancer metastasis happens, and we think it would explain a lot of the things that are happening in this area. This is something that is close to my heart. I have some family members that have had cancer metastasis into bone, so we're particularly enthusiastic about finding some answers there, and that's one of the reasons why we're doing this. This is where we're going with this. This was another project that I participated in. It includes 4 different organs on a chip. So the idea here was to create a body on a chip. This was the first one that we participated. So it included the liver, the heart, the lung, the vasculature. We would create these as modules that we would open and bioprint the vasculature and the liver inside. That's the work that I showed in some of my first slides. And then the team continued with this work after I left Harvard, and they created, this body on a chip with 2 organs for a first start. It was all interconnected. It was hooked up to biosensors and other things. And basically that's where we're going next. There has been other examples of that, and it's an area that is expanding. So with that, I'll finish off. And I think the question that I usually get asked more often is, why aren't we using printed organs already? Well, a number of reasons. To print an organ such as a heart or a liver, it takes literally billions of cells. This is hard, it's expensive, it's difficult to keep that many cells alive. The biology there is poorly understood. So as I said, right, the cost and the degree of difficulty it's one thing to put cells in a petri dish. It's another thing to understand the interactions of these billions of cells in a tissue. We don't know the long-term effects of these engineered systems. We know that they're not as good as the native natural ones. So, once you implant these, there's a lot that needs to be learned. There's a lot of regulatory and ethical concerns still around this idea, and the list goes on and on and on. So when are we going to do that? If not now, when? So this is a prediction from a paper that was published in Nature recently, and it shows, two-dimensional tissues can already be printed. There's skin examples out there, things that are already being used. Hollow tubes, we've printed some of those. Other people have printed some of those. They're already there. They're not regulated yet for clinical use, but they are available. Hollow organs, they're easier than solid organs, but we're also not there yet. Maybe in 10 years or so, we would be getting there. In solid organs, maybe 15 years or so. So these lists changes every time that I give this talk, because every time that a new paper comes out, you basically have to scramble, and things are moving a lot faster than you would think. One could argue actually that the idea of the solid organ, effectively, it has already been printed. You saw the beating heart, right? It wasn't exactly as the native beating heart, but it was an example of that. So you could probably shorten that timeline by a few years or so. So with that, I'll just thank some of my collaborators, some of our funding sources. If you have any questions, or if you're curious about the stuff that we're doing, or if you want to come visit the lab, spend some time with us, we have a lot of partnerships with UFC. Just visit the website, send me an email, and we're always happy to interact. So thank you very much, and I'm happy to take your questions at the end of the presentation section. Thank you.
Professor Bertassoni, thanks a lot for your presentation. It was, again, it's awesome, the work to develop. So congratulations again. And we have some questions already, but we're going not to ask you right now. So please, I would like to ask you to wait a little bit. So we're going to have Professor Mira presentation. So after that, we're going to have the discussion. So thanks a lot.
My pleasure.
So Professor Alex Mira, I know that we have this issue regarding your time zone. So thanks a lot for your comprehension.
That's fine.
So now I would like to invite you to present about so I'd like you to perform your presentation. So when you're ready, please.
Okay. Yeah. Can you see my screen now?
Yes. So as we would say, so this is about the microbiome, the last human body organ. So again, we'd like to thank you for your kindness, for your collaboration with our graduate program, with Professor Lidiany and all the students involved. So please welcome.
Thank you. It's my pleasure. Don't worry. Normally in Spain we go to bed very late, so I'm used to this, and that's not a big problem. It's actually for me an honor and a big pleasure to be here today. So thank you very much for the invitation. I'm going to talk to you about these billions of microorganisms that we have inhabiting our body, and I think we are more or less used to consider all bacteria and so on as being pathogens, but the truth of the matter is that many of them inhabit our body in the skin, in the gut, in the oral cavity, in the respiratory tract, in the vagina, in the eyes, and they perform many important functions for our health. And the last years has been really interesting and entirely fascinating to understand really what they are doing. And I have this subtitle, second title for the talk, which is The Last Human Organ. And that's because I consider all these microorganisms that inhabit our body as, another organ. It's not from a physical organ, like a heart or a lung, but it is really from the point of view of the functions that they can perform, as I'm going to show you later on. So as we have seen today, the number of bacteria, fungi, viruses, and so on are huge. In fact, between 1 and 2 kilograms of our body weight appear to be formed by bacteria. Believe it or not, there are people that have taken the time to measure this and to weigh, the weight of bacteria in our body. We know that at least we have the same number of bacteria as our own human cells. Well, there's also some estimates saying that it could be larger. It depends. For example, if we consider red blood cells as human cells or not, but in any case, at least the same number of bacteria as human cells. And then when we look at the number of genes that they encode, it's about at least 100 times higher than our own human genome. So imagine the large amounts of functions that they can perform for us. So I think that when you look yourself in the mirror now, you can consider that we are at least half human, half microbial, right? Okay, this is an example that I think illustrates well what I mean by being another organ. This is part of the work that has been performed by Bob, one of our PhD students, and he has been working on some oral bacteria that are part of our normal flora that appear to produce nitrite and nitric oxide from the nitrate that we ingest in our diet. Nitrate, NO3, is very high in many green leafy vegetables like lettuce, spinach, and so on. It's very high in beetroot, for example. And the thing is that our body doesn't have the enzymes to transform nitrate into nitrite. So this chemical reaction can only be performed. We depend entirely on our oral microbes to be done. Once they transform the nitrate from the food into nitrite, then we can swallow it, and in the stomach it can be transformed into nitric oxide, or these bacteria themselves can actually take it further and produce nitric oxide. And nitric oxide has a wide range of functions in our body. For example, this is a very important vasodilator, which means that your blood pressure goes up when you don't have these bacteria doing this function. So they are very important. And of course, then I first thought, well, how long are those bacteria going to be in contact with the nitrate from the diet? Only for a few minutes while we are chewing our food, right? But most of it is going to be swallowed and then it's going to be lost. But this is where things start to get really interesting, because our body, during human evolution, has found a way to recycle a big part of that nitrate that goes into the bloodstream. And then when the blood goes near the oral cavity, the salivary glands are going to actively capture the nitrate and to concentrate it into saliva. And as a consequence of that, about half an hour after you have ingested food, we have a peak in the levels of nitrate in saliva, and the levels can be 10 times higher as the concentration that we have in blood, for example. So our body is really making sure to provide nitrate to our oral microbes to perform this function. So that's why I say that they are like another human organ. It's so much integrated into our physiology that we make sure that we provide food for these organisms to, in exchange, obtain nitrite and nitric oxide as a factory. So this is the way we test it in the lab. So for example, we ingest a glass of beetroot juice, which is rich in nitrate. After some time, it is recycled in the blood, and then it goes, it's pumped by the salivary glands into saliva. And there, our oral microbial factory makes the job and transforms the nitrate into nitrite. And the nitrite in our body gets transformed into nitric oxide, and then we have a vasodilation and our blood pressure can go down. These are actually real data from 2 individuals. This is the levels of nitrate that we measure in saliva, and as you can see, half an hour after the ingestion of this beetroot juice, we have a peak in nitrate. Then we swallow it, but then it gets recycled again and keeps coming back, and therefore the levels of nitrate can be quite high for 5 or 6 hours. Here we have another individual that doesn't recycle the nitrate so well, so this varies between individuals. And then when we measure the levels of nitrite, so that's NO2, the product of this reaction, we see a peak a bit later. So about 1 hour, we see that the nitrate is being transformed into nitrite. And then again, the levels of nitrite stay quite high for a few hours. Here we have another individual where that perform multiple oral mouthwashes with an antiseptic. So you kill not only the bad bacteria in the oral cavity, but also the good ones like this. And this individual that has killed all their oral microbes is not able to produce nitrite because we depend entirely on the microbes. You know, if a few years ago people would have told me that our bacteria in the oral cavity were going to influence our blood pressure or our cardiovascular health, I would have thought that they are crazy, you know, but that's what we are learning today. Okay, so what do they look like, these microorganisms in our body? Well, in many cases, as we listened in the previous talks, they are forming biofilms. So they form an association with other bacteria in these complex biofilms. This is an image performed with the latest microscopy technique called classy fish, by which we can individually label different organisms, each of them with a different dye, with a different marker. So in the past, we could do this with 2 or 3 bacteria, no more, because then the spectrum, the wavelength of the markers would overlap. But now with these new techniques and image processing and so on, it can be done with 10, 20 organisms or more. So this is an image that was performed by people at the Forsyth Institute. And it is a dental plaque, which is the biofilm that forms on your teeth 24 hours after toothbrushing. So this is the way that your teeth or your biofilm in your teeth looks like if you don't brush your teeth for a day. And each color here is from one bacteria. We have 10 different bacteria marked in this sample preparation. And the area I'm marking here on the left is what is attached to the surface of the teeth. And the area here on the right is what is exposed to the oral cavity. And the first thing that really calls our attention is that this ecosystem is dominated by this organism that we know very little about, which is called Corynebacterium matricioti. We didn't know almost anything about this, that forms these very long filaments that run radially from the base to the edges here. And it's really like the main architectural feature of the biofilm. And then at the edges of the filaments, we have marked in green here all Streptococci. And Streptococcus is a bacterium that is able to survive in the presence of oxygen. And it forms this kind of shield around it to protect the inside of the biofilm from the oxygen. And therefore, it allows the formation of an anaerobic environment here in the middle, where strict anaerobic species like these ones marked in white here, like Fusobacterium and others, which are strict anaerobes, actually are able to survive. So this is much more structured than we thought. And this is in the oral cavity, but of course we think that the same thing could be happening in the gut and in other areas of the human body. So they are organized and they appear to be close to friends, and we are starting to understand how they interact. I mean, when I look at this beautiful image, it reminds me of an Amazonian rainforest in Brazil or something, but at a microscopic scale. I think it's absolutely wonderful. And we didn't know that this was the way that they looked like until a few years ago. Okay, another thing that we didn't know is we're actually more than half of the bacteria that we have in our body, because we just have not been able to culture them. So this is a phylogenetic tree, a tree of life of all bacteria and archaea, the main group of unicellular organisms. And its name here is a phylum, so it's a big family of bacteria. And all the ones which are marked in white are those phyla for which not a single bacterium member has been cultured to date. And in fact, microbiologists are so special about it that you need to culture a bacterium in a pure culture in order to give it a name. And this is why all these ones in white, are called like OP3, OP10, TM7. I mean, TM7, we didn't know it existed in the body until a few years ago. And then we found it's very common in the oral cavity of people with gum bleeding and gum disease. And we didn't even know it existed until the DNA sequencing methods allow us to know that they are there. So imagine the big change that we are undergoing now in the field, and especially by the use of the so-called omics techniques, by which we can, for example, extract the DNA of a sample, and then by PCR amplification or by direct DNA sequencing, we can look at all the organisms who are there, who is there, what genes do they have, so what can they do? Or if we want to have a more functional approach, we can extract the total RNA, then we sequence the total RNA. That's what we call the metatranscriptome of a sample. So we can look at all active bacteria in the sample, for example, and what genes they are expressing at a particular time point. This is fantastic because we can know what they are doing, And the same if we look at the total metaproteome, or all the proteins which are being produced at a particular time point, or the metabolites that are the product of the activity of the microbes. So all these techniques really are giving us an extraordinary image, about what they are doing. Of course, a big problem with this is that we obtain millions of sequences and data, and actually we need really to have in our team to have some people experts in bioinformatics and so on to process all this information. And I always encourage all the students who want to work in microbiology to actually have some basics about Linux, or you don't need to program, but at least to be able to use all these wonderful tools that we have to do the analysis of the microbes, because the combination of experimental and analytical skills will make you more employable in the future. This image here in the left is not from our lab. I took it from the internet because it looked a bit more cool. And this one that doesn't have the glamorous view of this one is actually what we have in the lab. So it's a computer cluster with 250 processors. And therefore, if we want to compare all the sequences we have against the databases, if we do it in a normal computer, this would take for a few months. I can tell you because it happened to us. And now what we do is that we split the work, we share the work in parallel among all those 250 nodes, and therefore that computing, that process that would be computationally very heavy can be done just in a few hours. So I always say that this is the kind of life I thought I was going to have, looking at the microbes under the microscope and culturing them in agar plates and working in the bench like this. And although we also do that, most of our time we spend our life as an oral microbiologist or like this, studying the sequences of DNA, RNA, and proteins and so on on the computer. And I know this might look a bit boring, but I'm going to try to convince you today that that's not the case at all. We are really in the middle of a revolution in the field. And I think that if, all the pioneers in the field, like Luiz Pasteur and company, they would be really amazed and very excited to be living specifically this period of scientific research in the field of all the microbes that we have in our body. As an example, to illustrate this is a work that we did a few years ago where we looked at the bacteria in blood after the dentist had extracted the wisdom tooth. If this happened to you, you know that sometimes you are given even antibiotics, and that's because a big hole is open in your oral cavity, and some bacteria can invade from the oral cavity into the bloodstream, and that can be dangerous. They could even go to the heart and so on. So how do we know if there are bacteria in blood? Well, this is done by culture. Even today at hospitals, you take the blood sample and then you culture. And in order to know which bacteria we had in those blood samples after the tooth extraction, what we did was to use all the possible culture media that we had. We cultured them also with and without oxygen, trying to culture everything that as much as we could. But we could only identify or isolate 4 bacterial genera. The most common was Streptococcus, that we know that it's considered to be, the most dangerous or the most common in these kind of cases. The size of the word here is related to the proportion with which we found that bacteria in the cultures. But then with the other half of the blood sample, we extracted DNA and we really looked at all the bacteria that were present there. Not only those that we could culture, but all of them. And that's what we found. So we were really just looking at the tip of the iceberg of this enormous biodiversity that we were missing, because in the culture we favor the growth of some bacteria, but we entirely miss all the others. And not only that, but we not only had a very incomplete view, but we also had a very biased view, because, for example, the Streptococcus that we thought was the most common bacteria, if you look at here, it actually occupies a very small proportion of the total. So imagine that now, all what we have gained, all the insights that we can get. Imagine that we can get samples within a cavity to know what bacteria are involved in the development of dental caries, or we can get a sample and analyze the bacteria in people with inflammation in the gut, or what happens in the lungs of people having or not having invasion of pathogens. Anyway, this is just an extraordinary moment to be looking at all this because of the new techniques. Another important point I want to make is that every part of the body have their own organisms. This is a statistical approach that we call a PCA, Principal Component Analysis, and every point here is a sample that we have analyzed. We have analyzed the DNA to know which bacteria there are in that sample, and then the samples that are clustered all together means that they have a more similar bacterial composition, and the farther they are from each other, more different bacterial composition. We did this initially to look at whether the bacteria that we find in breast milk, that also has lots of bacteria, are actually a contamination from the skin, but they are not. You see that all bacteria from the skin cluster here, oral samples cluster here, samples from the vagina, breast milk, and the gut. So you can see that even if many of these environments in the human body are linked with each other, for example, we swallow 1 liter of saliva per day, and many bacteria in the oral cavity are going to go into the gut, but the bacteria in the gut are entirely different from those in the oral cavity. So each environment is different and is going to select for those ones that adapt specifically to that niche. So if you give me a sample now and you don't tell me where it comes from, I can analyze it and I can tell you, okay, this is a sample from the gut or from the oral cavity or from the vagina. Although sometimes we do find bacteria in places where they are not supposed to be, right? But after some time, things go back to normal, and then you find what you should on each environment. Another interesting thing is that many environments that we considered or we thought in the past to be, completely or almost as sterile, actually have many bacteria under normal circumstances. I think we all know this bacterium, Helicobacter pylori, which is the causing agent of gastric ulcer. It can also cause gastric cancer, and it was thought that it was the only bacteria that was able to survive in the acidic conditions of the stomach and so on. But that's not the case. It is true that when Helicobacter is present, it dominates the niche. So between 70 and 80% of the bacteria are Helicobacter because they expand. But in the absence of Helicobacter, in individuals without Helicobacter, the diversity is much higher. So we have many bacteria which are not present there, including, for example, some lactobacillus that we have been able to culture and that appear to have an antagonistic effect against Helicobacter. So that's one of the other functions of our microbiome. They are able to protect us from invasion by pathogens. I think we all know, for example, that when we take antibiotics, we could have infections by Candida or any opportunistic fungi in the oral cavity, in vagina. So that's why we need those bacteria. One of the functions they have is to colonize the niche and to prevent, to keep a balance with our body and to prevent the invasion by pathogens. Another important studies that I think can be done with these new techniques is actually to study the bacteria we have under health conditions. So for example, take a sample from dental plaque of teeth which are unaffected by dental caries, or then take samples from the cavities themselves, from the initial caries from the outer layers in the tooth, or from dentin, the tissue underneath enamel, or at the deep dentin. This is work that was performed by Aurea Simon, who is a dentist that was working in the team. And what she did, she realized that the more that you sequence, the higher number of bacteria that you identify in a sample. But then after, when you sequence a lot, then you have reached already the saturation of the system, so you identify all bacteria present there. And this is what happens when you look at a healthy dental plaque from unaffected teeth, no cavities or anything. You see there are between 500 and 600 species in those teeth. But then when you look at the different cavities, especially the initial cavities, the number of bacteria goes down a lot, only to 100. That's probably because the environment is very acidic, and then only those bacteria which are able to survive under those acidic pH conditions are able to survive. But still, the number of bacteria was very high. And in the past, 100 years ago, it was thought that Streptococcus S mutans was the causing agent of dental caries. It was isolated from a cavity of a patient. It was able to synthesize the glucans, as we saw today in the wonderful talk that we saw. They are able to transform sugars into acid, so it lowers the pH, and that what actually makes our tooth to lose the mineral. But they are also able to transform the sugars into this sticky, glue substance that they use to form the biofilm. So it was thought that S mutans was the main causing agent of dental caries, but we identify here more than 100 species. Of course, when we first published this, people told us that perhaps many of those bacteria are not doing anything. They just happen to be in the cavity, but they are dead or they are not doing anything. I always compare this with the construction workers, at least in Spain. People always say that in a construction site, if you have 5 people, only 1 is working or 2, and the others are watching, And I think this is quite international, apparently. So if we extract the DNA here, we would think that all of them are actually doing the work, whereas we need a trick to identify those which are active. So that's what we do by extracting the RNA, which is the active part of the genetic material. So that's the metatranscriptome. We extract the RNA, and then we look at which bacteria are there present. And this is what happens when we look at different cavities. These are dentin cavities from different individuals. And as you can see, the proportion of different bacteria is very different. So first, we confirm that there are many bacteria at each individual cavity. But to make things worse and more complicated, it really varies between individuals. So this cavity, almost 3/4 of the bacteria which are active is this one, which appears to be absent in this other cavity, which is dominated by this one, and so on and so forth. So that means that it's going to be very difficult to identify a single causing agent, and that is going to make it very difficult to control this disease, because it appears to be a clearly polymicrobial disease. It doesn't follow the general standard rules of having a single causing agent for an infectious disease. This is the most extreme case that we found. Not only cavities from different individuals, but cavities from the same individual, and this is extreme because it is from the same tooth. This is a molar. We extract the RNA from 2 different cavities from the same tooth, and we see that the composition is also different. So, in my opinion, this is the main reason why a vaccine against dental caries is never going to be efficient. It's not going to work. And in fact, for decades, many scientists have been trying to develop both active and passive immunization strategies, trying to develop a vaccine or directly expose the oral cavity to antibodies. And then when they do clinical trials, it never works well. Why? Because they are using as their target, again, surface antigens from Streptococcus mutans, And where is S mutans here? Well, I'm going to show you here is this thin line in here, which is about 1% of the total of bacteria that we find in cavities. And in fact, in about half of the cavities, we didn't find a single S mutans cell. So when it is present, it can cause caries, but it's not the only bacteria. And when it is present, it can only account for 1% of the total. So even if we develop antibodies against S mutants, the other group of bacteria can also produce acid and to develop or expand the cavity. So that's why a vaccine is not going to work, and we need something else. For example, we need to restore that balance, and that's why I believe more in the prebiotics and the use of probiotics, as I'm going to show you later. Another important concept that I think I want to transmit is that many of these of the pathologies that we find in the human body in relation to the alteration of the microbiome is actually not the consequence of an infection, but it can be the consequence of what we call it dysbiosis, which is a change in the balance between bacteria and the host. This is an example for gum bleeding and gum disease. As we see that the pockets, that the tissue that actually holds the teeth, as they get deeper and deeper pockets, which is when the disease is actually getting worse, we have an increase in these pathogens that we know are involved in the disease. They induce inflammation, gum bleeding, and it could even lead to the destruction of the tissue that holds the teeth. But I want you to look at what happens in the healthy individuals. I think that we always need to do this. What happens in healthy individuals that appear to be immune to the disease? And if we take these lines down to here, where the periodontal pockets are below 2.5 millimeters, they are not even pockets. In that case, they are considered to be healthy gingival sulcus. In here, it's not that they are absent, so they are present. All these pathogens are present, but they are below 1%. So they are at very low levels because they are kept under control by a healthy environment. But it doesn't mean that we are infected with them. I always say that we all have the enemy at home, So we have all these pathogens already. And when we have a change in the environment, a lot of sugar in the diet, or our immune system doesn't work well, or we don't brush our teeth very well, so the plaque starts to get bigger, and that causes inflammation of the gingiva. And even when there's a change in the environment, then they start to increase in proportion, and then they get transformed into pathogens. So that's why I think that oral diseases, for example, and other kinds of maladies that we have in the gut, like a lot of cases of gut inflammation, it's not because you get infected, but because there is a disbalance, because we have a dysbiosis. There is an increase in some bacteria which are associated with disease, but you don't get infected. It's not like drinking a glass of water that contains Vibrio cholera and then you get cholera because you get infected with organisms that you didn't have before. Here, we need to keep the balance, and we need to think about that framework if we want to design new strategies to control these dysbiotic states. Another important point I want to transmit is that many of these bacteria, fungi, and so on have an effect not only at the site where they are, but they could also have a systemic effect. We have an example here in the oral cavity where many bacteria that cause inflammation in the gingiva and gum bleeding and so on can also have a systemic effect. For example, they can affect our cardiovascular health, or even if during the pregnancy the mothers have this gingival inflammation, there is also an increase, for example, in the preterm birth rate. So some of these organisms, they produce inflammatory molecules, or they can even travel in the bloodstream or part of them, and that could trigger the birth or too early. So this is what we knew until a few years ago, but the last years has been unbelievable because they have shown to what extent these oral bacteria and gut bacteria and so on can have an effect in the rest of the body. This is data which is not from our lab. This was done a few years ago, and they did studies of post-mortem brains in people that died and that had Alzheimer's. And they did a staining for gingipain. Gingipain is a toxin which is produced by some oral bacteria, especially the ones causing gum bleeding. And this is when we make this staining in the gingiva of patients with gum disease, and you see that it is very much marked in red. That means that there is a lot of this toxin, of this gingipain, but in here, this is in the brain. So we have this layer of gingipain that happens in the brain of people with Alzheimer's. And to make things more interesting, actually the location of that gingipain is at the same sites where the alpha amyloid plaque is going to be placed. And you know that this amyloid plaque is actually what is causing the disease. There are some groups that have found that amyloid plaque have antimicrobial properties, and some people believe that it might be actually a defense from our immune system to fight against infections, and one of them could be this oral bacteria that could be there. So our oral health can have an influence not only in the oral cavity but also in some other parts of the body. We have a second example here with a Fusobacterium, which is an oral bacteria that appears to be in some cases in the gut. And when it is in the gut, it can actually cause no more and no less than colorectal cancer. So there are experiments in vitro where actually the presence of Fusobacterium, it causes a chronic inflammation, and it produces some toxins that are able to transform healthy cells into tumor cells. This would be the second case where a bacterium has been found to be able to induce a kind of cancer. The first one would be Helicobacter in the stomach, and then this would be the second one. In fact, 3 years ago, there was an experiment where they treated mice with antibiotics. It was an animal model for colon cancer, and the use of antibiotics eliminated the presence of Fusobacterium, and it was able to completely arrest and stop the development of tumors in these mice. So this has opened a lot of research in the field, but I don't want to scare you with all the the bad things that our organisms can actually do if we don't take care of them. But there are also many positive aspects, of course. I told you before that bacteria can prevent the invasion of pathogens, but they can also synthesize very important compounds. In the talk, before we saw that they are able to synthesize vitamin K, for example. And here we have another example, because some bacteria in the gut are able to transform the fiber from the food into short-chain fatty acids. And they have incredible functions in the body. They mediate the immune system. They can affect the release of insulin, and they can affect also our brain. They can affect. They can have an effect in memory, stress, in anxiety, and things like that. And then there are a lot of studies, mainly in animals, but many of them now also in humans, and the results are spectacular. So I have here some bacteria that are able to synthesize products that we are not able to synthesize. Actually, it's funny because many people believe that it's very important for our health to eat a lot of fiber, but we always think that it is because the fiber helps us in the intestinal transit, right? That it helps us to go to the toilet, basically. But actually, another very important function of the fiber is that some bacteria in the gut transform the fiber into these short-chain fatty acids that have very important functions all over the body. Another example of compounds that can be produced by bacteria are all these long list of neurotransmitters that can be produced by bacteria. We have here dopamine, serotonin, GABA, and so on. So this is part of what is being called now as the gut-brain axis, because many bacteria can influence not only the whether they can help us to digest foods and so on, but they can also have an effect in our behavior. And I think we all probably have felt that it affects our mental state when we take antibiotics and we completely eliminate our gut organisms. And now we are starting to understand why this happens and what can they do to interfere, and how can we use them as life organisms to improve our health. Of course, they can also eat, for example, or transform part of the drugs that we ingest. And we know now, for example, that there are some patients that do not react well. They don't respond well to chemotherapy, for example. And that depends also on the bacteria that you have in the gut. So we have some bacteria, for example, that have these enzymes which are able to transform part of the medicines, of the drugs that people have when they have chemotherapy, if they have cancer. Then they transform it in something else, and that means that they are not going to respond well to the treatment. And in other cases, what they do is that they have enzymes that transform some of the drugs into other compounds which appear to be toxic to the individual, and therefore those patients do not have a good time. They have a terrible time with the chemotherapy, because their gut organisms are actually going to produce some toxins based, using as a base the drugs that we give these patients. And therefore, more and more people are thinking about doing a test of your bacteria in the gut before choosing what is the best chemotherapy treatment for you specifically. Okay, how do we acquire all these bacteria, fungi, and so on? In theory, when the baby is born, is totally sterile, although now there are some teams that believe that the baby can be exposed already to some bacteria from the mother in the placenta, but that topic is very, it's not clear at the moment. So let's assume that the baby is born and hasn't been exposed to bacteria yet, and the first bacteria are going to be acquired when it passes through the channel of the birth, the birth canal. And that is a study that was performed now 10 years ago, and they took samples, fecal samples from the baby after they were born, and they compared to the composition of bacteria from the mother. And this is very interesting. This is again one of these PCA analysis plots. So the samples which are close to each other are more similar in bacterial composition. And when the baby was born by vaginal birth, the composition of bacteria in the gut, the fecal samples from the baby, are more similar to those in the vagina of the mother. So this shows actually that those organisms in the vagina are the first ones that the baby is going to be exposed to. However, when the baby is born by C-section, as you can see here, the composition of bacteria in the gut of the baby is most similar to the bacteria of the skin in the mother. So the baby is going to have a bad start, so to speak, because it's not going to be exposed to the right organisms after birth. And you know that children that are born by C-section actually have statistically a higher rate of autoimmune disease, asthma, allergies, and so on. And it could be that one of the reasons is this lack of exposure of the baby at the right time to the right organism. So now they're starting to try to compensate for that by actually passing through a swab some bacteria from the vagina of the mother to the oral cavity of the baby if it has been born by C-section, in order to try to control for that. We also did some work following what happens in the gut and in the oral cavity of babies that we followed in Sweden for 7 years. We took samples 1 month after birth, 3 months, 6 months, 1 year, 2 years, 5 years, and 7 years. And then we extracted the DNA from the samples, and we looked at which bacteria there were in all those individuals. This was work performed by Maita Sitik, who is now at the Karolinska Institute after doing her PhD with us. And we saw that, of course, the babies that were born by C-section had a different set of oral bacteria, as it had been shown in the gut. But we also saw other factors that appear to have a much longer influence through time. And one of them was the period of time that these babies were breastfed. This was performed in Sweden, and all mothers gave some breastfeeding for 1, 2, 3 months, but some mothers actually did it up to 12 months of the baby. And what we see here is that when we compare the bacterial composition of children that were breastfed for 3 months only, compared to children that were breastfed for 12 months, we see that during the first 3 or 6 months, the composition of bacteria is very similar in the two groups. But after 1 year, and especially after 2 and 7 years, they diverge a lot. And the bacterial composition, 7 years after the baby stopped having breast milk, is very different from those that had no breast milk. This means that it has a very long-term effect. And that's because it's going to affect how the composition of bacteria and how that ecosystem is going to build up. This is a video of an immune cell in breast milk. And one of the reasons why this is happening actually is because breast milk is full of organisms. We have here, I'm marking here this organism that is moving, as you can see. And this is a human cell. So it's a human cell from the immune system in breast milk. And what happens is that at the end of the pregnancy, many bacteria that inhabit the gut and the oral cavity and so on is going to transfer attached to human immune cells by the so-called enteromammary route, and it goes all the way to the mammary glands, where they are released into the breast milk. As a consequence of that, there are a lot of bacteria in breast milk under normal conditions. So this is the way that our human evolution has found to transport or to transfer the bacteria from the mother to the baby. Of course, we all know that, you know, the baby is going to swallow, to touch everything around it and so on. But that, you know, doesn't make much sense from an evolutionary point of view, where the breast milk is going to inoculate the right bacteria at the right time into the body through the breast milk. So that's one of the reasons. It was work by Alba Boix in our group. She's now doing a postdoc in the US. And but there is another important point. So the breast milk contains many live bacteria that get transferred to the baby. Sorry, I'm a bit tired. So that's what we call a probiotic. A probiotic is you know, live organisms that when they are administered in the right amounts to humans, they can confer an advantage, a positive impact in our health. But we have something else which is called prebiotic, and a prebiotic is a substance that we ingested into the diet. It can promote the growth of some good bacteria. And this is actually what happens in breast milk, because it contains, apart from all the antibodies, defenses of our immune system and so on, it contains also breast milk or human milk oligosaccharides, HMOs. This is a kind of sugar that is very interesting because the baby cannot digest those sugars. So they are not for the baby. So the breast milk, why does it contain sugars that the baby cannot use? Well, because they can be used by bacteria. So breast milk contains these human milk oligosaccharides that promote the growth of some specific bacteria which are very important for the development of the immune system of the baby. Therefore, this is what we would call today a prebiotic, really, but it was already invented, you know, thousands of years ago by breast milk. So I would say that human breast milk is probably the first source of prebiotics and probiotics in the history of mankind. So, you know, these prebiotics work as a sort of a natural, like a natural fertilizer to promote the growth of some bacteria. It has been tested in the lab, but no one has actually looked at what happens in the oral cavity, and I think it would be very interesting to look at that. So in general, I think the development looks something like that. It's what people call an ecological succession. So at the beginning, some bacteria are going to colonize first, they change the environment, and that allows the settlement of new bacteria. And then with time, this is going to develop more and more complex ecosystem. If you have a fire here, which would be the equivalent of taking antibiotics when you are an adult, it takes about now a month and a half or so until we recover all the bacteria that we had before taking antibiotics. So our immune system already knows what are our own bodies, you know, and we accept them and so on. But however, in children, we have seen that's not the case. If we have a fire here, we take antibiotics before 1 year or 2 years of age, that's going to change these initial phases so much that you are not going to have ended up having a pine tree forest. You end up having something completely different years later because the immune system is still not formed. So that's why it is very important we are exposed to the right microbes from the beginning and take care of this initial stage of 2 years. Okay, another interesting thing is actually that can be applied is to look at what's the interaction between bacteria and the immune system. This work, it all began, believe it or not, by this cartoon, where you have some bacteria being hit by antibodies and others that are able to escape, you know, to evade the immune system. So this that looks like a science fiction movie, we managed to do by the use of a flow cytometry, which is a microfluidic system where we pass the bacteria one by one, and we can label the antibodies so we can identify and to quantify and sequence the bacteria which are coated with antibodies and bacteria which are uncoated by antibodies, the ones which are not recognized by the immune system. So we went back to these children that we followed for 7 years. We know which ones of them developed asthma, allergies, and so on. And when they don't develop these symptoms until they are 4, 5, 6, 7 years of age. But we went to the first samples that we had, 1 month old fecal samples from the gut of these babies. And we have here what we call the IgA index. The values here are positive if the bacteria appears to be coated with antibodies, or negative values if they are uncoated. And it's very interesting, E. coli, for example. E. coli, which is a very common gut organism, it appears to be mainly coated with antibodies in healthy children, but those children that years later are going to develop asthma, they, their immune system is not able to recognize this basic organism. And the same happens with other bacteria. So we have a profile here that can serve us to identify years before the symptoms happen those individuals that could be at risk of developing asthma. So I think this opens the question of we could use bacteria, and in this case bacteria, the interaction of bacteria with antibodies as a biomarker for an early diagnostic of diseases. And I think this could be extremely helpful, because if we know individuals who are under very high risk of developing asthma, then we can act very early with a vaccine or by other methods and try to delay that or to prevent that from happening. And that's one of the main applications of these studies of the human microbiome. And there are many people that believe that in the future we will have one of these microbiome tests, either in the gut or in the oral cavity, in a routine way, very similarly to just having the blood test. And if you have some bacteria that appear to be present in people with colon cancer, then you can take another check, or you could have some bacteria which normally appear in individuals with Crohn's disease or inflammatory bowel disease and things like that. So it could be a way of actually testing your general health state, and perhaps we will see that in the future in hospitals. And another application of these studies is actually looking for health-associated bacteria that we could use as probiotics to improve our health. We wanted to know why there are people that never get dental caries. There are about between 10 and 20% of the population never develop caries, never develop cavities. So we wanted to know what happens, why is it, and do they have any particular bacteria that would be able to protect them? So we took dental plaque samples from caries-free and caries-active individuals. We extracted the DNA, and then we sequenced the total DNA. So this was the first metagenomic study that was performed in the world like this. And so we can look at who is there and what are they doing. And we identified lots of organisms associated with disease and with health. This is very interesting because we had very high levels of what appeared to be a new species of Streptococcus. So we said, okay, we can culture this one. So we went back to those caries-free individuals and tried to culture this Streptococcus. And in fact, it was a new species. What was really interesting and entirely fascinating for us is that when we culture this bacterium that appeared to be in healthy individuals, in caries-free individuals, next to bacteria causing dental caries, then we saw inhibition rings around it. So this is our anti-caries organism. Here we have a lone culture of S. mutans, S. sobrinus, so some bacteria causing dental caries. And you see that this organism forms like a shield because it is able to produce antimicrobial peptides that diffuse in the agar and they prevent the growth of the pathogen. This was, as you can imagine, extremely exciting in the lab, because we finally have shown, that people without dental caries have good bacteria that are going to protect them from the pathogens. So we said, okay, why don't we culture this, and then we can put it into a yogurt or in a toothpaste, and to try to incorporate into the oral cavity of the rest of mortals, that have dental caries. And it was a new species for science. We had to give it a name in Latin. We called it Streptococcus dentisani, which means healthy teeth in Latin. It's this beautiful bacteria here, and we perform all the different studies. And this is the way it kills S. mutans. So this is S. mutans here, one of the causing agents of caries. And after being exposed for 15 minutes to S. dentisani, and I'm going to mark you here these little holes. You see a pore there, another hole there, another one there, another one here, there. So this is the typical mode of action of antibacterial peptides, which are some sort of natural antibiotics produced by bacteria to kill each other, really. And S. dentisani has many of them, and it uses these to kill all these pathogens. And after they have all these holes, the organism is not able to keep the osmotic pressure anymore between the inside and the outside of the bacteria, and then they start to lyse and to burst. And this is what happens to S. mutans afterwards. You have S. dentisani here, and S. mutans here is releasing of the inside content because they are being killed by S. dentisani. So this is how bacteria kill each other. And this is what we think is actually happening at the microscopic scale in the dental plaque of these caries-free individuals. So I has been very excited. Actually, the whole work, began more than 10 years ago, because one of the women in our lab was one of these 10% of the population that never had cavities. And we were once having lunch, and she said, I never had dental caries in my life, but my boyfriend used to have lots of caries before going out with me. But after we went out, together, he stopped having cavities. And then I thought, okay, perhaps in the kissing, in the saliva, she is passing him some bacteria that could prevent. So she was our first patient. We took samples from her, and we isolated this bacterium. And then now we have found it in between 99 and 100% of the population who are caries-free. So very important who you kiss, And then we try to develop this. So first we did animal trials, and all the safety measures that we needed to do until they allowed us to do some clinical trials in humans. And we apply it in dental ferrules like this for 5 minutes. And what we see here is that the comparison with a placebo, where we only have this gel, but without the presence of S-dentisani, in comparison with the placebo, we see that the application of the probiotic induces lower dental plaque index, so lower accumulation of plaque, lower gingival index, lower inflammation, a more neutral pH, and other features that I don't have time to talk about, but we are very enthusiastic. This is the work that was performed by Marian Ferrer and Arantxa Lopez in several clinical trials. And we are very happy because we think that we have here an organism that can be used as a probiotic, and I hope that is going to be in the next few years in the market, because I think it could really help us to improve our oral health. So, of course, it is not enough that it's actually able to improve our health. It also has to be able to be produced at large scale. So we started making them in a small 1 liter flasks in the lab. This is 100 ones in another institution, and now it is being made by over 1,000 liter fermenters in Austria. And so now the cost of each dose of S-dentisani is very low. It's just a few cents of a euro, of a dollar. And we think that this is going to be possible to take into the market. So our idea is to have one dentisani that could be used by the dentist to put it into a varnish in all teeth at high densities and so on, and another cheaper treatment that people could take either in chewing tablets or toothpaste or something on a regular basis. Okay, so this is an example of how the basic research that we do can actually end up in having a product that can actually improve our health. This is more or less a summary take-home message about the work I have talked to you about today. Several approaches that we are using can actually give you a better picture of what is going on, the culture, the sequencing, the omics approaches, and so on. We can use some bacteria as a marker of disease. We can use the bacteria that we have in our gut or in our oral cavity as a biomarker to actually predict the disease. And this perhaps could be applied in hospitals. I think that the use of prebiotics and probiotics can improve our health, and especially in the oral cavity, for example. I think that's really the key point, trying to keep the balance, to restore that balance that has been lost by the dysbiotic state by the use of these compounds. But of course, we are killing a lot of our microbes by the overuse of antibiotics, by the Western way of life, the change in diets, that not being exposed to the microbes, having our food extremely clean and so on, and that's going to have an effect in the future, especially for all these kind of conditions which are affected by the immune system. And of course, something that we knew from the point of view of love, but now from the point of view of science, that kissing can also improve your health, at least your oral health, and of course, always if you kiss the right person. So I want to thank the people from my lab. This is the way that we have to meet during the last years because of the pandemic, but now we are going back to normal life. I hope that this improves in the future, and we can continue doing the clinical work, and I wish you all the best, and thank you very much for the invitation to be here. I'll be happy to take any questions.
Hello, Alex. Congratulations for your presentation.
Thank you.
Very nice presentation. So now we are going to have the questions. I think we have some questions. I would like to apologize to you and the other presenters, because besides being a periodontology professor in our graduate program, I'm also in the international office. So I'm going to host another room right now. We are hosting someone from the Canadian government for talking about international affairs. But I'd like to congratulate all of you, congratulate Professor Lidiany for bringing up this idea of this webinar. I think it was a great success. We had almost 100, actually have now 100 people who had viewed our webinar. It's going to be in our website. And I'd like to, again, to congratulate you all, and thanks a lot for your presentations. I'm sorry and apologize because I'll have to leave you, because I'll have to start the other one in 10 minutes. Thanks a lot.
Thanks, Rodrigo, for your presence here. And I emphasize your words about the congratulations. The speeches were amazing. Thank you, Alex. Thank you, Simone. Thank you, Luiz. It was a great moment for us, especially discussing so timely topics. So we have some questions here. Let me search. The first one was to Simone. And Hamili asked, how important is the formation of the acquired pellicle in in vitro caries models? Should we always perform this step regardless of the substrate being enamel, dentin, composite resin, etc.? What do you think?
Thank you, Lidiany. Thank you, Hamili, for the question. So that, as I mentioned, choosing an assay is important to understand what your questions are. So the salivary pellicle, we know that it happens, it is exposed to the oral cavity without a biofilm on it for a very short time, either when the baby's born, the teeth actually is born, and the teeth erupts in the baby, or when we do a professional cleaning, right? So in this case, the only reason why the salivary pellicle would be important is for this initial formation. Once the biofilm is established, that is not going to play a role anymore. But if we think about a product, so if we are working with a product and we want to know how the initial biofilm formation is with those first colonizers and cell membrane and receptor interaction, then it's extremely important that you have the salivary pellicle, because that's probably where the receptors with affinity for those membranes will be. But otherwise, if you are studying something and you want to see the long term, then I don't think it would be necessary. So it's really based on what you want to see. So yeah, if you're looking at as you mentioned, a resin or a material, then it might be important, because that relationship between the material and the pellicle might be important for the first, for the initiation of the biofilm. I think, don't know if that answers the question, but
okay.
That's
I think that is that. Do you want to compliment Alex? Something?
Well, I was going to say that that perhaps after toothbrushing, you eliminate also most of the biofilm but I don't know to what extent you eliminate completely all bacteria or not. But perhaps it might also play a role after toothbrushing. I don't know what do you think, Simone?
Yeah, you're right. Depending on the access you have to the surface, right? But you don't eliminate the pellicle, though. The pellicle will be there. But every time you're looking at the initial phases of biofilm formation, I would incorporate the pellicle.
Mm-hmm.
Yeah.
I guess that the next one can be done to Simone and also Alex is related to biofilm models to produce artificial carious lesions in dentin. And the question is related to the best microorganisms to be used in this type of model. What do you think about it?
Do you want to start, Alex?
Okay. I mean, it's very difficult. I think that in general, I personally prefer not to use a combination of bacteria that we select, because that's going to be very artificial. But of course, you can also control better what is happening. So you could have only a single species biofilm with H mutants, and perhaps it can be valid to test many aspects of the biology that we want to address, because it's going to attach, it's going to produce acidic, so it's fine. But of course, that's nothing to do with the real world. So I actually prefer to take a fresh sample and to inoculate it into our system. That's what we call a microcosms experiment, where we actually try to mimic really all the complexity of the composition of bacteria that we want to study. So I like that a bit more, but it is true that you cannot control very well all the conditions, whereas if you have a 1, 2, 3 species biofilms, then it's easier for you to control that. So I think it depends, in my opinion, on the study that you want to test and the questions that you are addressing.
Yeah, as you just mentioned, there is always pros and cons, right? So the microcosm, the problem I see is that by culturing it, you were selecting bacteria and you don't know what exactly you were selecting. So again, it's pros and cons, right? If you look at the lesion, so whatever you have there that produces the lesion you want, that should be enough. But maybe we should, I always say that we treat caries as the same disease, but maybe we should think about it, because is it the same? Enamel caries, that's why I kept saying that the models I have are mostly for enamel caries, because that's where that acidic environment will play a role. But when you go to caries in dentin, that's a different process. So, and honestly, I don't think we have a model that is good enough for dentin caries. We just translate the model we have for enamel caries to dentin. So maybe that's an open opportunity there for people to really look into models that will really work on different caries, because the process is different. So yeah, that's my...
Absolutely. I fully agree. And then in dentin, we find lots of bacteria which are able to degrade the collagen, for example, that appears to be present in the dentinary tubules. And they can even survive in the absence of sugars, because they are able to take the sugars from the dentinary tissue themselves. So they have adapted to be independent of external sources of nutrients. And it would be an entirely different model. And I fully agree with you. In relation to the, when we take a real sample and culture it in the lab, what we have done is to sequence what we get, and then also sequence what we obtain after it grows in the lab. And of course, it's very much influenced by the culture media that you use. In general, if you put any sugar, you're going to have an increase in streptococci in relation to what you have in the renal sample. But by changing a little bit the culture medium, you can more or less try to mimic and to grow a very similar ecosystem to what you have in the real world. But it is, of course, every single culture medium that you use is going to have an influence. So yeah, again, I totally agree with you that you have to select the right question and then to choose what is the right model to answer your particular question.
I have one question for Luiz. Do you have any experience in using bacteria or microorganisms in organochips, or in some model that you use tissue engineering?
Yeah, we have before us, other people have done co-cultures of mammalian cells with different types of microbial species, especially in the gut and in other areas where they are a vital part of the physiology there. And what we have done recently, we published a paper in JDR a couple of months ago, which actually addresses some of the comments that you guys were making earlier. We developed this tooth on a chip where we have the dental pulp cells on one side and a biofilm kind of like on the cavity side of the chip. And we haven't published that particular experiment. We have some, I think, maybe supplementary data on that in that paper, but we can co-culture the two at the same time. So it's basically, it's a organ on a chip model of the tooth that replicates kind of like a dentin cavity, right? So the biofilm is on the surface of the dentin. We can see the bacteria traveling through the tubules, and the dental pulp cells are on the other side. And then we treated that with MTA, and we're looking at how, right, MTA or other types of silicate cements are able to just deplete that biofilm, which we saw. But where we're going to next is to understand how the biology of the biofilm interferes with the biology of the pulp cells in the cavity through these, what we call paracrine, right, like communication. So it's like soluble components that are being secreted by the biofilm and byproducts of the degradation of the dentin matrix that we know travel down to the tubules, interfere with the biology of the mammalian cells in the pulp, and then we can address these two independently. So we're kind of like recreating a cavity on a chip, and that's one of the things that we've been looking into. Yeah, I mean, it's not an active project right now in the lab because we've been doing other things on other organs, but we've definitely been interested in replicating that in the tooth too.
Very interesting. And do you remember the bacteria that you used?
So this was work that was done with Michel Kuh from UPenn. He's the microbiologist person in the project. I'm not. That's why I just keep quiet when you guys are talking bugs. But I think that we've cultured both S mutans as a single species biofilm. We've loaded a microcosm biofilm as well onto the chips. It's actually easier to culture them. We generated quite a bit of preliminary data on that. And Michel has done some co-cultures of S mutants with some candida as well, because he's interested in some of that, I guess, interaction. And the good thing about the platform is that it's agnostic to the type of cell you use. So you can have, presumably any type of bug, and you can culture these. You can culture these cells independently. You can keep the biofilm exposed to one type of medium, the mammalian cells to another type of medium, and you can keep it under flow. So you can really see the interaction between host microbiome interactions, right? That's really, I guess, what guides a lot of the things. And it's the reason why we did that is because I think it's fair to say that this is what drives a lot of the biology that happens in these complex systems. Is not, you rarely will have just bugs or just mammalian cells. You usually have an interaction of the two. And it's probably naive for us to think that they don't talk to one another and that they are not interfering with, right, each other's microsystems. I'm sure they are. So hopefully with that model, we can start dissecting more of these interactions.
Great, thank you. I have another question for you, but I'm not sure that you are able to answer because I'm not, I don't know if it was related to a specific slide. But the question is, can these be used as scaffolds for bone grafts, assisting in optimizing vascularization? Do you think that the vascular system would be recognized in vivo by the patient? I guess that was related to the vascular system that you showed.
Yeah, I think that we're actually beyond that point. We don't use these as scaffolds technically anymore. We actually make the tissue entirely, right? So technically, the concept of scaffold starts from the principle that you would load a material, and then the cells can kind of like move into that material and slowly process that scaffold into an actual tissue. What we have been doing is we build the tissue with all of the characteristics that native tissue have in the body. So, and we've done this, you know, like through multiple characterization methods, we can ensure that we match the nanoscale structure, the physical properties, the genetics, the protein level expression of the cells that are embedded in these systems. That's essentially what we have in that Nature Communications paper. And now when we implant this engineered bone with the vasculature in the body, we haven't done this in patients, obviously, we can't, we're not allowed yet, but we've done this in animals, in multiple animal models, in different models, calvario bones, subcutaneous, and others. And what we find is that the blood vessels that we make in the lab, they connect exactly with the host vasculature, and we know that because we tag them with different colors, right? So our engineered vessels are green, the human vessels or the animal vessels are red. We also use human cells, and we know that animal cells are from a different species. So then when we put them together, we can clearly see that they're fusing, and we also see blood going through. So we know that our engineered vasculature anastomoses with the host vasculature in vivo. And we've also seen that we have complete regeneration of a critical size of bone defect in a question of 8 to 12 weeks. So our engineered bone presumably replicates exactly what happens when you have autologous bone, right? Which is kind of like the real gold standard. It's not even BMP, it's actually autologous bone. You take it from the iliac crest in a large defect, you pack it into the patient's defect, and that gets remodeled over time. We're about to publish a paper where we show exactly that remodeling happening, and it's very similar to the autologous bone. So to answer the questions is, yes, it happens. We can load it into the body. We see the processing of our engineered tissues with the cells, and we see the connection of the engineered vasculature with the host vasculature as well.
Great, thank you. Sarah Guedes asked for Professor Alex. This was a great lecture, since you have emphasized the microbiome as the last human organ. How do you see the use of antimicrobial agents in dental caries and periodontal disease? I guess that Simone also can help in answer.
Okay, well, I mean, I think that we would never take antibacterial antiseptics to swallow to sterilize our gut on a daily basis, like we do, for example, very commonly in the oral cavity. So I think actually I am, of course, in favor of using oral antiseptics and oral mouthwashes at specific time points if you are having gum disease and you are going through a particular period, or, I mean, of course, on those particular cases, I think they are good and they have to be used. But what I don't think is a good idea is to entirely destroy the oral ecosystem by the daily routine, daily use of oral antiseptics as part of our routine oral hygiene measures. We know, for example, that when we brush our teeth, that improves the ecosystem because it actually favors those bacteria which grow in the presence of oxygen and so on. And that's true for the tooth and also for the tongue. When we remove, with a toothbrush, the biofilm in the tongue, that also improves the quality of the ecosystem that we have there. We have an improvement in Rothia and Neisseria, some bacteria that we know reduce the nitrate and keep a healthy ecosystem and so on. So the physical removal by toothbrush is good. However, the total sterilization by antiseptics on a daily basis, I don't think is good. And we are starting to understand part of the outcomes of that. One of them, for example, is that your blood pressure goes up, imagine. So I think that it's actually the point of view today that most people have is not to completely eliminate the biofilm, but actually to try to drive the composition of the biofilm, try to encourage the growth of those bacteria which are beneficial, try to reduce the activity and the proportion of those which are harmful, but not to fully eliminate it. That's my point of view.
Simone?
I agree 100%. And even in situations where we have to remember that everything we put in the mouth, if we don't swallow, we spit on the sink. So that means antibiotic going to the water system. So that can cause so many other issues that are not just related to our own body, but related to our planet. So that is also something to keep in mind. I think there is very, very few situations, and that's why I mentioned in my talk, and I think if we have to do something to treat, the best is to do it site-specific treatment, because then you are really only like treating what you need and not necessarily the entire mouth. There are ways to control caries, and that you don't need to use antibiotics or antimicrobial drugs. So just to add that point that we sometimes forget that we have an important mission with our planet, so we have to be careful.
I would also like to add something else, because Luis has mentioned Michel Kuh, and he's doing a wonderful job also by the use of nanoparticles that are, so I don't know, I would call them intelligent nanoparticles that contain the antiseptic on the inside, but it's only released if the pH gets acidic. So you have them, you could ideally have them there, but the antiseptic is not released unless the pH goes below 5.5, and then it opens up and releases the antiseptic. So you only make use of that when you really need it, and I think that's a very clever strategy, and we might actually see more than that in the future.
Okay, thank you. And the last question is for Alex also. Do you believe that infection by SARS-CoV-2 can change the oral microbiome, even if temporarily, for a short period of time?
Well, what has been shown is that it has changed a lot by after the lockdown, because we were at our places without having interaction with our human beings for many months, and that lack of interaction really has lowered the number of organisms that we have in our oral cavity, gut, and so on, by lack of interaction with other people. But I'm not sure that by the presence of the virus itself, we could have a change in the microbiome. What has been shown is that depending on the bacteria that you have, you could have actually a worse or better outcome of the disease. So people with gum bleeding and gum disease, for example, they have a higher levels of inflammophilic organisms and organisms that actually induce inflammation at the systemic state, and therefore people with gum disease at a higher level of periodontal pathogens in the oral cavity, when they get infected, they have a worse outcome. So higher death rate and longer, days of stay at hospital and worse symptoms. So I think this is one of the factors, like if you are obese or diabetes, or you have some cardiovascular problem, all these diseases have in common that you have a general inflammation state at the systemic level. And that's also true for the oral cavity. So you have gum disease, you have more inflammation in the body, and that's a risk factor for having worse symptoms of COVID-19.
Thank you. Dear friends, I'm very happy to be here, and I'm very honored to have this amazing afternoon and evening for you, Alex. Many thanks also.
No problem.
I know it's late. It's late there in Sweden, okay?
Yeah, I'm in Sweden. In Spain it would be better, but here, people have dinner at 6 p.m. So my neighbors might be now going to bed, but it's fine, it's fine for me. No worries.
Many thanks for all of you. Many thanks. UFC is very proud of to be you here. Okay? Muito obrigada, gente. See you.